Can you all see my screen okay? I get the nod from Jessica, thank you. All right, well, great, welcome. And yeah, for those who didn't hear, just sign on, like there's a bit of a power outage happening here in North Seattle. So I'll do my best to facilitate as usual, but just heads up that we may need to be scrappy and adaptive over the next few hours, but hopefully things will be stable. And I'll just go over quickly what the plan is. We meet quarterly, so this is our fall meeting, and I've come to enjoy it as a time when we get to reflect on what we've just seen this last year, the last field season. So we'll have some quick updates in the middle of our agenda, where we can maybe do a little group thinking about what happened for each marine mammal species in our purview. But luckily, we're going to start off with some longer presentations and kind of zoom in more than we have for quite a while in on harbor seals. So I'm excited for both of those. And if we have some time left at the end, we'll do some housekeeping. We actually didn't meet in the spring, partially due to COVID and other complexities, but we had a plan for 2021, and I think we were going to approve it in the spring meeting. So hopefully we can approve that and it begins some brainstorming about 2022 work plan. And then if we're not all completely zoomed out and folks want to, we'll stop the recording and just have some social time because, you know, COVID. Yeah, I'm looking forward to some time when actually meeting together. We used to do sort of regional meetings. So down south in the fall and around here in the winter, and then up north in the spring to try to give everybody a chance to meet in person. But I will tell you, it feels like a long time since that has happened. So given that a couple of you are new to these calls, let me just go over some of the infrastructure we've set up, which was pre-COVID, but it's turning out to be helpful when you're trying to do asynchronous collaboration and communication. So just really quickly, let me run through my standard introduction. We have a roster, and it's currently the most useful form is a Google Sheet, and you're welcome to hopefully comment on that. If you see your name on there or you don't see your name, you'd like to be on there, you can add your contact list, contact information. And that's sort of an evolving thing. We're trying to standardize on the PSEMP roster format as well. But you might want to just check that to see that you're on there. We also have a web page. So if you Google PSEMP and Puget Sound Partnership, you'll find this page where there's a box note. It's kind of the top level of the Marine Mammal Work Group. So that's a place where you can find a link to the agenda document and the history of past meetings, as well as some of these other resources. Historically, we've used the Google Drive to share documents. And so in this shared MWG folder, you'll find some species-specific work group content, and some of that we'll go over today. And then we also have these subgroups, which are pretty well populated right now. So this is another sheet. It's kind of a different view into who's interested in what within the work group. So this is organized by species, with the high priority ones in the white zone here. And I hope we'll eventually get to these gray ones, but we've been making progress over the past few years in adding expertise across these different species. And so you can be a lead and actual member of one of those subgroups. And there's been volunteer lead authors on various work products, but there's also ways to be a little less involved, like just say, I'm interested in proofreading anything related to California sea lions. And then if you're interested in joining those groups, but you're not sure, just put your name in this column and I'm happy to reach out to you and give you a sense of, you know, who's doing what and how to collaborate better. Yeah, so related to those subgroups, I've been trying to build distribution lists for email. So we have a general email listserv, but been rolling out over the last we're really just during COVID trying to make these other distribution lists useful to each of those groups. We have a Facebook page, which is pretty quiet, but I'm trying to set it up along with other channels for doing better about publicizing any work products that we generate. All right, so just segueing into recent news and announcements, and I'll cue you at the end here to, if anybody else has anything to add, but these are what's been on my radar. This fall, the State of the Sound 2021 was published, so you see a link to that. Unfortunately, Andrew wasn't able to organize a COVID BC symposium this year, as he was also not able to do last year. So that was canceled, but hopefully it'll happen next fall. There was earlier news, which you may have missed, because really the last time we touched on this was back in January, but I had taken some of these notes in anticipation of our spring meeting. So there are a few other reports that are relevant to some of our work groups, sub work groups. So I'll just link those for now. We just finished what was a great Acoustical Society of America meeting a little more than a week ago. And Marla and others in Canada presented quite a lot of recent southern resident killer whale data, particularly from DTAGs. There's also a movement model that's pretty much ready to be operational up in Canada for southern resident killer whales. So I think you'll be seeing some more publications, but that I hope some of that information will come to the forefront in the next couple of meetings through Marla and others. As Emily can tell you, there were also some great, some cross-pollination across humpback researchers from Hawaii and here, but I'll just leave it at that for now. But I think everybody is seeing changes, interesting changes in the humpback acoustic behavior and occurrence patterns. And then most recently, I learned about something called the Pulse that I wanted you all to be aware of. There's a new NGO up in Anacortes called the Salish Sea School, and they've lined up folks like Stephanie and others to give some talks every month in 2022. So some of those I've flagged as relevant to us. So just take a look at that if you're interested. There is going to be a Salish Sea Ecosystem Conference in April, all virtual. So that'll be followed by yet another COVID delayed event, which was supposed to go off this month, but now will be next July, August in Florida. Anybody else have recent events or news to share? All right, good. I guess I'm mostly got the pulse. Great, so I think we should take a little less than 10 minutes now to just quickly go over some general PSEMP updates. It's mostly me sharing news, but feel free, Katrina, maybe you can jump in on point 4 here. So, PSEMP is a longstanding effort that really grew out of PSEMP back in the late 80s. And various work groups have come and gone over the years, but two that had become quite quiet and inactive were the spatial data group and the modeling group. And both of those have spun up again this year. And I've attended a couple of their meetings, or at least watched notes from their first couple of meetings. And I'm personally interested in both of these, but my impression is that the spatial data group, and at least initially, is focusing mostly on riparian mapping. But I think it's noteworthy that there's a groundswell of interest around that, and in part because there's been a growing need to share transboundary southern resident killer whale location data better. And I think those two we may have an opportunity to kind of leverage what they're building in terms of infrastructure with what we've also been building for southern residents with the Canadians. So I just wanted to flag that. And if you're interested in ecosystem modeling, that group is also spinning up. The other thing that's happening in the grander scheme of things is we're beginning to implement the revised vital signs. And that's in the scope of work for many of the different work groups, but one of them is marine waters, where they're considering a noise indicator, which would be relevant to multiple marine mammals. And so they've started thinking about that formally this summer, and both Rob Williams and I gave them presentations in the late summer. So I think there'll be an opportunity for us to interact more formally with the Marine Waters Group. And Aaron has volunteered to give a presentation on some of their efforts to, funded by PSEMP, to generate sort of a foundation for monitoring noise in Puget Sound. So we'll hear more about that, both from Aaron and Rob, and also from the Quiet Sound folks. So there's interesting work locally happening with Quiet Sound, quiet commercial ships, that relates to noise monitoring. And there's also growing global initiatives to standardize how we measure noise levels in the ocean. So anyways, I think that's going to be an exciting topic for 2022. Have anything to add to that, Katrina, or do you want to walk us through this bullet point 4 in whatever way you see fit?
So I want to quickly touch point back around, Scott, that you noted around the Quiet Sound initiatives, and especially noting that since Lynn and Todd are with us. So either Lynn or Todd, if you have updates or would like to provide input, please feel welcome to do so, but no pressure. I can just quickly dive into the PSEMP and partnership related funding opportunity that just occurred this last biennium, and going into this next one. So hopefully you all have seen the Monitoring to Accelerate Recovery. We're really excited to announce a series of projects that received funding that help support the vital sign and PSEMP programs. So like Scott noted, Aaron's work around monitoring noise is supported through that funding opportunity, but there's a series of others that may be of interest too. And just noting that that announcement was also paired with the collaborative Puget Sound Scientific Research funding opportunity that was also hosted by the partnership, and has a series of projects that have a strong overlap with this marine mammals work group. So just want to flag that, and Scott did a great job of just noting some of those projects in the agenda. Let's see, I think I'll leave it there, and just noting that, again, there's a lot of great efforts coming up the line.
Super, thanks for that synopsis. And I'll just add that Nathalie has offered to give us a presentation on some of those successful proposals at our next meeting. And I'll invite, there were a variety of proposals from other marine mammal work group members that weren't funded, and I think they deserve a chance to talk about what I think were some pretty inspiring ideas that could be funded, or useful collaborations outside of the PSEMP and Puget Sound Partnership funding opportunities, which will come back around in 2023, right, Katrina? Or assuming that the state funds them.
Yep, that's exactly our hope.
Great. All right, so let's move on. It's just about time to queue up the presentations about harbor seals. So we'll aim to take the next hour or so to try to work our way through these 4 presentations, starting with Tom, has he arrived yet? Okay, great.
Yes, I'm here.
Good, I see you up there, great. Would you like to share your screen, Tom, for this, or do you want me to advance your slides for you?
Let me try to share my screen real quick here. I haven't done this in a little while, so hopefully it's going to work okay. Oh, it says I cannot share my screen while other participants are sharing.
Okay, so I just stopped.
Okay, let me try it again then.
Yep.
See if it'll work this time. I think it's going to work this time, it looks like. Okay, can you guys see my PowerPoint title slide now?
Looks good.
Let me make it full screen. Can you see it as full screen now?
Yep, looks great.
Okay. All right, well, thank you very much, Scott, and thank you, everybody, for being here. This is my first attendance at this particular group, so it's a pleasure to be here, even though it's only virtual. Hopefully someday I'll get a chance to meet all you guys in person. I've met some of you, but a lot of you I haven't met in person. But for now, I guess while COVID is still with us, we're just going to have to do this virtually like just about the rest of the world. So anyways, we do what we need to do. So I'm going to talk today about estimating stock size of harbor seals in the inland waters of Washington using line-transect methods. And this is a PSP and Long Live the Kings funded project. And my co-authors are Mari Smultea, Eric Ward, and Barry Barajikian. And we have published a paper on this, which I'll show you a screenshot of the title page towards the end of the presentation. So you can see on the upper left here some of our funding sources. The initial fieldwork was funded by the Navy and by NOAA. The analysis work that we're presenting here was funded mainly by PSP and Long Live the Kings. Okay, so a little bit of background. Harbor seals in inland Washington waters were, of course, reduced by predator control programs in the 1900s, as were pretty much all the pinnipeds and some cetaceans as well. However, the good news is that stocks clearly have rebounded quite a bit since protection in the 1970s in Canadian and US waters. The past stock size estimates have used primarily more standard pinniped methods of counting seals on haul-out areas during times of year when the maximum number of animals are considered to be hauled out, and then developing a correction factor for animals that might have been in the water during the time that those surveys were made. And unfortunately, the most recent published stock size estimates are about 22 years old from 1999. So there's no recent information to update this in the published literature, although I know that there is a new effort underway to update these estimates that I think we'll be hearing a little bit about later on. So our analysis was intended to sort of try to fill in a gap here of a needed more recent population estimate. And as probably you all know, there are 3 management stocks recognized by NOAA: Southern Puget Sound, Hood Canal, and then the Washington Northern Inland Waters stock, which includes the Strait of Juan de Fuca and San Juan Islands, but only the US portion of that area. So a little bit about the context. As I mentioned, generally pinniped estimates are made using primarily counts of hauled out seals with a correction for seals that were in the water. And basically, there have been several previous in-water estimates made from line-transect survey data from our earlier analyses that we did of harbor seal numbers in the water, but unfortunately those did not provide an estimate of overall population or stock size because they didn't include an estimate or a correction for the number of seals that were on land. And they only included seals that were in the water during the time that the surveys were done. So our approach here was to basically investigate how we could use the data that were collected on line-transect surveys, which only counted or surveyed seals in the water, but use those data and develop correction factors so that we could get full stock size estimates for the 3 harbor seal stocks, which included all the animals present, which includes, of course, in water and on land animals, on shore animals. So the approach was basically to reanalyze our aerial survey data and refine our Puget Sound harbor seal estimates of density and abundance from earlier work that were done for Navy reports, incorporate these new data to try to get these correction factors, primarily using dive time data from tagging studies, and produce these new estimates of the size for the 3 harbor seal stocks. And of course, then to write up a paper for publication and to present the results at various conferences and meetings, including the one that we're doing today. So this project, of course, is applicable to understanding a bit more about harbor seal impacts and predation on salmon and other protected stocks of fish in Puget Sound, among other things. So a bit about the methods. I'm going to go through these pretty quickly. They're all covered in the report or the publication. Briefly, the surveys were not specific to harbor seals. They were focusing on all marine mammals that were present in the area. They primarily were set up with the focus on harbor porpoises. So the survey lines were designed primarily to make sure that we got good coverage for harbor porpoises. But as you can see from the map on the right, the survey lines were also pretty well set up to give representative coverage of the whole area. So they should be useful for allowing us to estimate estimates for other marine mammals as well. We flew systematic transects using a fixed wing aircraft. There was a total of 38,431 kilometers of survey effort conducted. We observed a total of 4,678 harbor seal groups, so we had a pretty large sample size. And we filtered the data to only use data collected on systematic transect lines in low Beaufort conditions of Beaufort 0 through 2, and where there was no hard glare. And so that restricted our effort to about 8,040 kilometers of effort and 2,437 harbor seal sightings. In addition to the line transect surveys, we incorporated information from a separate but concurrent study that was done in the same general area, tagging harbor seals for working on other issues. And again, we use these seal tagging data to develop our correction factors for animals missed on our survey. The tagging data used satellite link time depth recorders and fast lock GPS tags. The tags were glued to the back of the seal. And we use these data to develop this model to estimate the proportion of seals that were diving and thus missed on our aerial surveys, and also the proportion that were hauled out at the time that we conducted our surveys to get full stock size estimates. So this is the standard set of line transect equations. I'm not going to go through these in any real detail. This is pretty standard stuff in terms of how line transect estimates are done. But again, I wanted to mention that we did filter the data so that we only use data collected during systematic transect lines and what we considered to be good sighting conditions, which included Beaufort 0 through 2 conditions and situations where we didn't have any hard glare that would really restrict our ability to detect seals. So moving into the results, harbor seals were sighted in all parts of the study area. You can see in the map on the right here, the blue dots are our harbor seal sightings. You'll notice here, first of all, that the number of dots showing in the northern area of Strait of Juan de Fuca and the area around the San Juans is much lower than in the main part of Puget Sound proper. And that isn't an indication necessarily of lower densities there. This is just a distribution plot. And our effort was much lower in this northern area than it was in Puget Sound. So don't interpret this as a lower density of seals there. That's not what this is intended to show. It's just basically showing where we detected our seals and the data that were available to us for conducting the estimates. As I mentioned, after filtering the data, we were left with 2,437 sightings. So even though it reduced our sample size quite a bit, we still had quite a large sample size for doing the line transect modeling. Maybe move on. So again, looking at the tagging data, we use these tagging data to develop sort of a equivalent of, if you're familiar with line transect analysis, what we call a track line detection probability, or G of 0. But in this case, unlike with cetaceans, the G of 0 estimate that we produced had two components. One was the typical number of animals that were missed on our survey because they were diving underwater and were not available as our aircraft flew over. That's a pretty standard G of 0 component. But in our case, we had another component, and that was seals that were hauled out on land during the time we did our surveys, because our surveys were focused again on the water. We weren't doing haul out counts on land. And so we needed to develop a correction factor for those seals that were on land. So we used the tagging data and used it to develop these proportions available, which includes those two components that I just mentioned. And the results are shown here on the left by week of the year. So week 22, I think that's in the springtime sometime, going through week 40. And this shows the estimates of the proportion available by time of day. So you can see there's generally a peak in the middle part of the day here. And the estimates are higher in the spring and lower in the latter part of the study period. The G of 0 correction factors range from a low of 0.249 for the autumn season or fall season to a high of 0.429 for spring season. And so we combined these correction factors with our line transect estimates, which gave us estimates of the number of seals in the water. And this series of graphs on the right-hand side, the three graphs here, shows the detection probabilities for the three stock areas here. And you can see that they're very, very similar. These are three different stock areas using three different sets of the data. And the shape of the curve is almost exactly the same for all three of them. So that was good to see that we had consistency there. After truncation of the data to develop the best possible estimates of abundance with the highest level of precision, we were left with 2,353 sightings of seals to be used in calculating the three detection functions. And again, there was, as you can see here, there was a separate detection function calculated for each of these three stock areas. The final results in each case used a half normal model with a cosine adjustment. And overall, we feel like we achieved a good fit for all three stock areas with similar detection curves. So moving on a little bit to our final take-home message, conclusions. After developing the correction factors from the tagging data and applying those to our line-transect estimates for number of seals in the water, we came up with estimates of the entire stock size for each of these three stocks. The Hood Canal stock, we came out to 1,368 seals with a CV of about 17%. The Southern Puget Sound stock, 1,976 seals with a CV of a little over 20%. And the Washington Northern Inland Water stock, 7,513 seals with a CV of about 11.5%. So I'm going to move on here. Comparing these estimates to previous estimates that were available. So as I mentioned earlier, we didn't have any really relatively recent estimates from the 2000s. Everything was basically from the 1970s through 1999 that we had available to us. But there's obviously undoubtedly been an increase in the number of harbor seal numbers in these stocks since the 1970s when protection for these animals was put in place. But there has been evidence that, in fact, there's been some stabilization and maybe even possibly some decrease in numbers in the mid to late 1990s. And this is, thought to be related to the fact that the stocks may have been reaching a level at or near their carrying capacity. So our estimate that we came up with, if you add all three stock estimates together, you get an estimate of 10,857 seals, which is a bit lower than the previous estimates that had been available from 1999 of about 13,700 seals and 1993 of about 16,000 seals. So moving on to management implications, this is sort of I suppose it's not really quite accurate to call it a novel approach because other people have used something similar, but it's certainly an uncommon approach to estimating density and abundance estimates for pinnipeds, using line-transect data for in-water estimates and then correcting for animals on land. So it's a bit of a preliminary attempt at this, but it provides a much more recent stock assessment for the three NOAA stocks than the ones that were available from 22 years ago. And so these stock estimates, it's critical that we have obviously, timely estimates of abundance for these stocks because it's important for evaluating impacts of harbor seal predation on threatened fish stocks in Puget Sound, which obviously is a major goal of this group, this organization. It's also important for quantifying impacts of anthropogenic activities on the seals themselves. And further, it also has implications for understanding potential reasons for the relatively recent increase in big killer whale numbers in Puget Sound, which I think we're going to hear more about later as well. So I mentioned earlier in the talk that we have published the results of this study. The paper was published in PLOSONE. It was published on June 9th of this year. And this is a screenshot of the title page of the paper. And if anybody would like a PDF of this, I think it's available from the PLOSONE website free of charge. But if you want me to send you a PDF of it, just pop me an email and I'll show you my email address as well as Mari's in a bit here. So talking about the status of the work for this project, we're basically, as you can see, complete for all aspects of it, except for the final aspect, which was to present Puget Sound Ecosystem Monitoring Program Mammal Working Group presentation. So I can't quite say I'm complete yet because I've got a few more words to say, but as soon as I'm done with my talk and answered any questions, I can consider the last one to be complete as well. So upcoming next steps for us in terms of kind of follow up on this work is we have just been awarded funding to conduct a similar type of analysis to estimate numbers for both California and Steller sea lines in Puget Sound. And we're just getting started on that right now. We're really in the very preliminary stages of that. But I hope that I will be able to present results, myself or Mari will be able to present results on that study to you at some point in the future, maybe a couple years down the road. And we also do have plans to publish the results of that paper in a reputable peer-reviewed journal at some point in time as well. So just to conclude, again, I mentioned that if you would like a PDF of the paper, my email address is here. Mari's email address is here. You can write to either one of us and request a PDF of the paper. And I just wanted to mention a few real quick notes here of thanks to our funding sources. Puget Sound Partnership and Salish Sea Marine Survival Project were the main funders for this analysis. The initial survey work, line-transect survey work, was funded by the US Navy through NAVFAC and also through Pacific Fleet. The Northern Waters aerial surveys were funded by the National Marine Mammal Laboratory, and the seal tagging study was funded by the state of Washington and NOAA Fisheries, as well as other in-kind contributions. And so with that, I think I will finish. And I don't know if we want to do questions now, or if you want to just move ahead, Scott, and maybe take questions later.
Yeah, let's do one question now. And I'm trying to buy Cindy Elliser some time. So in anticipation of buying her a bit more time, Casey, would you be willing to go next? And we'll put Cindy at the end of these four.
Sure. Yeah, that'd be fine.
Okay. Yeah. So any burning question from anyone? I see a hand from Lynn. So I guess you win. You get the one question.
Oh, great. Hi, Tom. Lynn
Hi.
Barry, No Fisheries. How's it going?
Good.
I'm just wondering if you have done any kind of crosswalk with the methods that Jeff Lake was using for the same type of analysis to estimate those three different inland stocks as well as the Washington coastal stock, and if you could speak to any differences you might be aware of there.
Yeah, we knew that Jeff and Steve Jeffries and other folks from NOAA and Washington Department of Fish and Wildlife were working on updating their estimates that were, the last estimates were available again from 1999. We had known that since the beginning of the study, but the timing of availability for the information for our report came in a little bit too late for us to really include any mention of that in there. I understand that there is a paper in the works now, which I think we'll hear a bit more about later. We did receive a very draft version of some of the analyses of that. But unfortunately, it was, like I said, it was kind of at the point where our paper was already in press. So it wasn't really possible to really do any detailed comparisons. But just briefly, of course, that analysis is using the more standard, typical way of doing pinniped population estimates by using haul out counts and then correcting for animals in the water. And I think looking at the preliminary numbers that they had seen in the report that they had sent us, it looks like their estimates were significantly higher than ours, especially for the northern inland waters area. And so once we have a full report from them and everything available to look at that, hopefully we can kind of take a look at those and see if we can figure out what might be going on there. It's certainly possible that our estimates, especially for the northern inland waters, might be a bit of an underestimate. And one thing that makes me think that that might be a possibility is because, as you probably remember from the slide where I showed the sightings, we had much lower survey effort in the northern inland waters area of Strait of Juan de Fuca and the San Juan Islands. So we basically had, I think, about 5 or 6 days of survey effort there in the spring. And we had, unfortunately, not very good weather conditions for some of the area, especially at the outer end of the Strait of Juan de Fuca. So those estimates, I think, should probably be considered fairly preliminary at this point. But I think that, if I remember correctly, the estimates for Hood Canal and southern Puget Sound were more in line.
Great. Thank you, Tom.
Thank you.
Really appreciate it. And yeah, maybe we can ask a few more questions as we proceed. You've reminded me to invite you all to, as I did in the email, to paste an update. If you're not presenting today, I'm very curious to know what everybody has been seeing and observing, hearing out in the Puget Sound on both sides of the border. So don't hesitate to do that, and we'll try to capture that in our notes. And similarly, feel free to keep your eye, if you're a presenter, on the chat because that's another place where folks can keep asking questions. So let's go on to you, Casey. Are you able to share your screen?
Let's see. I imagine I can, if I can figure it out. There we go. Well, yeah, thank you, Tom. Well, first of all, nice to meet all of you that I haven't met. This is also my first time at one of these meetings. And just want to say thanks, Tom, for presenting your and Mari and your co-authors' work. And with Lynn's question, that was about as good a lead into this stuff, which is the other stuff that kept getting referenced. And if I can make it full screen, go into presenter mode, I'm just going to update you all on that paper that Tom was mentioning that was unfortunately, given how timing works on these things sometimes, just wasn't ready in time for direct cross comparison. We did fly surveys, just a quick update on as well on what we've been doing. We did fly surveys this year, but the paper's already so far along and the analyses are done, they're not included here. So this is through 2019 for most of the stocks and only through 2014 through the outer coastal stock. But so for our paper, there's another little difference that makes it a little tricky to compare with Tom's, which is something I'll discuss at the end here, but we've actually proposed a new boundary, a change in the boundary between the southern Puget Sound stock and the northern inland stock. It was previously down here at the Tacoma Narrows, and that's based mostly on genetic work that was done a while ago, but sampling was so coarse for that we knew that down here was different than up here, and there was a line drawn. The Narrows is a nice sort of oceanographic physical feature to add in there, but we've got some reason to suspect it's actually probably a little farther north, which makes comparing our estimates for those two stocks, directly to Tom et al.'s a little tricky. But so these are some figures from our paper, which we're really hoping will be submitted here anytime soon. I started in my position in January and was told that we really need to get this paper out. We're still plugging away at that, but you can see all the black dots represent harbor seal haulouts in Washington State. And then these different colored dots zoomed in on each of these stocks is just showing in our last count in the area, roughly how many animals were there, binned pretty broadly. And so I was just going to present our time series and our newest results with the growth curves for some of these. This is the analysis that Lynn was mentioning that Jeff Lake had done, contracted for us. And so we do have numbers that are, well, it's a little tricky because some of our numbers are a little more recent, they're as of 2019. But our numbers in general were a little higher than the ones that Tom presented. And I think partly that's just different survey methods, and you can look back and try to get some more, direct comparisons, but the aerial surveys weren't flown in every year from our point of view either. But you can see this, as Tom was mentioning, a lot of these stocks, you'll see this pattern continue, have gotten to a point where if they're still growing, it's pretty minimal, or they appear to have approached carrying capacity a while ago. So for the southern Puget Sound stock, we estimated there's about 2,400 animals there. Looking at the coastal stock, we split this up between the sort of northern outer coast of the Olympic Peninsula and then the big estuaries, minus the Columbia River. That's always sort of been included. Most of the haulouts are technically in Oregon, and that's always been included on the Oregon side of things for reporting. And I think that ODFW has a paper in prep for their outer coastal stock, which will include the Columbia River animals. But you can see there's lots of animals down here. Close to 14,000 is our estimate. And that one's been sort of slowly growing and stabilizing over a while. Lots and lots and lots of animals in Willapa Bay and Grace Harbor. For the northern inland stock, a lot of animals up there. We have pretty sparse coverage, but it does look, other than bouncing around a bit, like things have stabilized. We break it up into the Strait, San Juan Islands, and sort of the eastern bays. We estimated about 16,500 animals as of 2019. And then Hood Canal is a bit of a puzzle. Hood Canal seemingly just bounces around and does not lend itself to fitting a growth curve, a logistic growth equation. And what we have done, fortunately, one of our last little tweaks before we submit this paper, we're hoping to incorporate we're going to incorporate some aerial surveys from that actually Cascadia that John did many years ago. And he just sent me the last little bit of data that we need And we're working on incorporating those. So that'll be a 1977 survey, another WDFW 1978 survey that we dug up, a couple in 84 and a couple in 88, which hopefully will give us enough of sort of a leading tail to actually capture some of that growth. But even in John's 1978 report on their harbor seal work, he had collected some anecdotal evidence that said that the harbor seals in Hood Canal had rebounded already really substantially by the late 70s, and that some people who lived in that area had said there were 3 times as many animals as there were a decade or 2 prior. So we may just be a little out of luck there. We may have actually missed the recovery period. And I'm actually intrigued by the fact that these numbers start out pretty high and almost decline a little bit. I think that we have to do some correcting and adding in of the data and plotting them all together. But with these new data, just eyeballing it, I think they actually do start out a little low, come up to a peak here in the late 80s, early 90s, and maybe drop back a little bit. And I don't know, I have a whole avenue I'd like to pursue of looking at how habitat on log booms around the sound has changed over the past several decades. And just looking at, a lot of people have talked about maybe we need to look at artificial haulout habitat and potentially reducing artificial haulouts as a way of controlling the harbor seal population. And I think we've done most of that already just by the reduction in the log booms that we used to have in Puget Sound and just don't really anymore, at least not nearly to the same extent. So I want to talk a little bit now about that stock boundary between that northern inland stock and the southern Puget Sound stock. I gave my lead in before, which is that it was fairly arbitrarily placed. I think some more fine scale genetic sampling could be really helpful here, but we get this pretty substantial gap in harbor seal haulouts just north of Seattle here, getting up towards Whidbey Island. And it seemed pretty natural to put the line in there somewhere, but we really also, we've collected a lot of anecdotal data there. The genetics do back up that line, but we also, I just recently made these maps of harbor seal tagging data. I think it's some of the same data that Tom et al. Used in their analyses, but this is animals from 2014 and 2016 that were tagged by WDFW, by Steve Jeffries. And the darker triangles are the places that the animals were captured and tagged, females on the left, males on the right here. And so the light blue are animals that were tagged in the northern inland stock, the green are animals that were tagged in the southern Puget Sound stock. And that little red line is where we're proposing to put the boundary. And you can see that the females, this is breeding season, so it's not surprising that they didn't wander too far, but they're nice and well behaved, so to speak, although crossing an international border. But it's very clear differentiation between these northern ones and southern ones. And I think there's 10 females in the whole sample size across the 2 years. For the males, it's not quite so clear. You can see that at least a few individuals crossed that boundary. So there were 15 males tagged, 10 males tagged over the 2 years. No, sorry, it was 15 males tagged over the 2 years. 10 of them never crossed the line. 2 of them had 97% or more of their locations were on the side that they were tagged. And then 3 of them actually did appear to cross and stay across, or spent half their time in one or half the time in the other. So maybe there's some more, to be investigated there. We're about to tag a bunch of seals up in Port Susan, up by the Stillaguamish River, so that should be sort of informative and educational. But what we're going to be presenting a sort of a tech memo sort of derived from the main elements of this paper at the Pacific SRG in spring, with the recommendation that stock boundary be moved. And they will then also consider Tom and Mari's work, and then consider our results as we publish them for their next stock assessment. So we've been working with Sharon Moline at NOAA to hopefully get new stock assessments for these seals, which are very much overdue. And I think all of the new attention that has been paid to harbor seals recently and to abundance of trends will really help to inform that. So we're going to keep flying surveys, but that's our little update now. And I don't know how long it took, but I'm probably close to my 10 minutes if I'm not over. So I'm happy to answer a question if we have time, or later if we don't.
Yeah, let's keep going, because we don't want to run into our break time too much. But I'll just remind folks, even if you're watching a presentation, if you hit the little chat bubble, conversation bubble down in the middle of your Zoom interface, you should be able to ask Casey questions. And there's a good dialogue going on so far, but it could even be more active. So let's thank you, Casey, and move on to Rebekah, if you're ready.
Hi, can everyone hear me okay?
Sounds good.
Awesome, thanks. I was having some connection issues earlier, and hopefully that won't be a problem this time.
Familiar. Yeah, we're seeing your screen. Looks good.
Okay, great. Well, hi, everybody. Thanks for having me here today. My name is Rebekah Lane. I'm a special projects contractor at Smultea Sciences, but I also have some other affiliations. I'm a program coordinator at the Marine Mammal Center in Sausalito, and I'm also finishing up my master's thesis at San Francisco State. But today, I'm going to be talking to you guys about the results of a paper that we just published last year titled Marine Mammal Occurrence, Distribution, Abundance, and Behavior in the Inland Waters of Washington from Aerial Surveys in 2013 to 2016. And so this is work that we did with Mari Smultea and Tom Jefferson as well. So just a brief intro, although I'm sure you guys are all familiar, but the Salish Sea is regularly inhabited by 10 marine mammal species, 3 pinniped, 4 odontocetes, and 3 mysticetes species. And there's at least another 15 species that are documented there occasionally. Climate change and reductions in prey availability and other human-related stressors are resulting in these rapid shifts in species distribution and occurrence. And so that being said, the comprehensive in-water surveys that provide us that baseline data in the region are pretty scarce or outdated, and particularly aerial surveys. So in Puget Sound, we have harbor seal, California sea lion, northern elephant seal, and Steller sea lion populations actually increasing and expanding despite their historical depletion in the area. Humpback whales are returning to the Salish Sea in the late 2000s, post being nearly extirpated from the area by commercial whaling. Small numbers of gray whales of the Pacific Coast feeding group are seen seasonally in Puget Sound now since 1996. Currently, harbor porpoises are the most abundant of the odontocetes in Puget Sound, and their population is increasing, whereas Dall's porpoises appear to be decreasing there. And of course, we have the 3 killer whale stocks that inhabit the Salish Sea, with the southern resident stock being listed as endangered. So our study area covered the inland waters of Washington State, including Puget Sound, Hood Canal, and the Strait of Juan de Fuca and the San Juan Islands. We determined our line-transect protocol to be consistent with previous marine mammal aerial surveys in the region. We have 8, to examine regional differences, we have 8 geographical subregions that you can see here to the right. There are 6 survey periods starting in August 2013, going all the way to January 2016 that span all 4 seasons. We did not conduct surveys over the 4 restricted air traffic zones that you can see here in red. And we recorded species, group size, numbers of calves and pups, observed declination angle and bearing to get more accurate latitudes and longitudes, behavior state, minimum and maximum dispersion distance between individuals, among other things. So we can get into results just a little bit now. It's a massive study, so I'm trying to kind of get through it as quickly as I can. So harbor seals were observed most commonly and year round. About 76% of our sightings were actually harbor seals, and they were primarily in northern Hood Canal, in southern Puget Sound, around Fox Island, west of Tacoma, and in Skagit Bay, north of Quano Island, in the South Whidbey subregion. Harbor porpoises were also seen commonly. That's about 18% of our sightings, and they were year-round, with the highest concentrations being in central and northern Admiralty Inlet and southern Puget Sound. California sea lions were observed in all seasons. Steller sea lions were only observed from fall to spring. And additionally, we had 2 Risso's dolphin sightings that are considered extraliminal. Both times they were just the pair of 2 individuals, and those sightings were made on August 31st and September 4th in 2013. This species was not observed in subsequent years. So results of our sightings and occurrence, you can see this map to the left here has sightings by species in the area. After filtering out for the survey conditions, about 3 out of our 10 species had sufficient sample size for density and abundance, which we'll talk about in a little bit. But yeah, I already did this. Sorry. So like I said, the harbor seals, harbor porpoises, and combined sea lion species were the only 3 groups that we had sufficient sample size to do density and abundant estimates. These are really preliminary, as Tom was saying earlier, because some of the prominent haul-out sites in the area were located in restricted no-fly zones. So this is really an underestimate of total abundance. High in-water abundance in Admiralty Inlet with about 15 sea lions compared to other regions. Our pool density of all seasons was about 0.0177 individuals per square kilometer. And then our abundance for the preliminary abundance for combined sea lion species was highest in spring with 66 individuals and lowest in the winter. So again, really preliminary because we were not able to go into those prominent haul-out sites, and our numbers would have been much higher. Group sizes for all species ranged from 1 to 150 individuals with a mean of about 1.8. Largest groups were the hauled out harbor seals. You can see in this picture here. Mean is about 14.5 individuals. The highest mean in-water group size was for killer whales, about 6.8 individuals per group, and then Dall's porpoises, and then harbor porpoises. But that being said, we had pretty low sample sizes for killer whales and Dall's porpoises, which I think 4 groups and then 2 groups. So 4 groups for the killer whales and 2 groups for the Dall's porpoise. So it's just, that's what we saw, the means, but it's pretty small sample size to make any kind of assumptions there. With group cohesion, it was highest for harbor seals, an average of about 6.5 body lengths, and California sea lions, and then Steller sea lions. And the harbor porpoise cohesion was highly variable, and we weren't able to really we weren't aware of any previous studies to kind of show cohesion in harbor porpoises and compare. Calves and pups were observed in 3 of 10 species: harbor seals, harbor porpoises, and killer whales. Harbor porpoise calves and harbor seal pups were most often seen in July and September. We did have a killer whale calf that was spotted with 5 other individuals on April 19th of 2015, and it was confirmed by a National Marine Fisheries Service research vessel and the Orca network as a group of transient killer whales with a small calf. We spotted another small killer whale calf in January 2016 with 8 adults, and it was again confirmed by the Orca network, but instead as part of the J22 group of the southern resident killer whales. For our behavioral observations, this is a map of any locations of milling, foraging, feeding observations by species based on the color there. So the most common observed behavioral states were rest or transit. Groups were sometimes seen milling, likely foraging or socializing. Active foraging and feeding was rarely seen, only about less than 2% of sightings, and it was only seen in harbor porpoises, harbor seals, Steller sea lions, gray whales, and then a minke whale outside of Puget Sound. Reactions to our aircraft were also noted, and they were pretty uncommon, 0.3% of our total of 10,412 individuals cited. And a possible reaction was recorded 31 times, and that usually consisted of an abrupt dive that was seen in harbor porpoises, harbor seals, 1 California sea lion, and 2 Steller sea lions. So a little bit of discussion about kind of how different, how we use this data to look at change in occurrence and distribution. For one, we did see that California sea lions were present in our study in all 4 seasons, and historically they were not recorded from June to October in the area. Harbor porpoise populations, like we discussed earlier, have clearly increased, as you can see from our data, whereas Dall's porpoise populations have decreased pretty significantly in the area. So during our aerial surveys, only 4 small groups of Dall's porpoises were seen in like 3,000 kilometers of survey effort in the northern waters. And reports of Dall's porpoises by the Orca network were limited to like 45 sightings between 2013 and 2020. So this indicates a clear decline when you consider that in a 1997 study, there were about 1,500 Dall's porpoises seen. So like we said earlier, our study did exclude one of the largest documented California sea lion haul-out sites in Puget Sound, since the surrounding space is a no-fly zone. Inclusion of this site would probably have increased our abundant estimates, definitely indicating a higher abundance than what was reported historically. And this increase in local abundance from the 1970s and 80s is expected, especially when you consider that both California sea lion and Steller sea lion abundance along the West Coast has been increasing in recent decades. Let's see. So our observation of gray whale feeding pits in the Snohomish River Delta, we observed foraging both actively and inactive in the event of these foraging pits that we saw. So this was consistent with previous studies that also saw feeding pits from gray whales in the same area. The rare reactions to our aircraft and results of other studies showing similar things support that this method is viable for studying marine mammal behavior. Our group sizes were consistent with all of those reported, except for our Risso's dolphin pair. Usually they're in groups of like 10 to 100. So obviously that was a pretty strange. The harbor seals, our harbor seals were 4 times farther apart than the other 2 pinniped species. There's no clear relationship between cohesion distance and group size for pinnipeds, at least not in our study. And like I said before, we don't know of any other studies of group cohesion of harbor porpoises. So we could only really compare to a study by Mari in 2016 that reported mean maximum cohesion distances among other odontocetes. And so harbor porpoises had a mean cohesion distance of 3.9 body lengths compared to 7.7 for Risso's dolphins, 5.3 for common dolphins, and 5.2 for bottlenose dolphins. Our data for calves and pups suggests that we have strong calving and pupping seasonality in harbor porpoises and harbor seals that both peak during the summer months from July to September. And clearly the observation of a killer whale calf in the J22 group, southern resident near Tacoma, is important considering the endangered status of the ecotype. Back to these Risso's dolphins, that year, or between November 2011 and April 2013, there were 32 sightings of Risso's dolphin, of a Risso's dolphin pair. Our photographs were sent to Cascadia, but were not high enough quality to confirm if these were the same individuals as the pair that had been seen 32 times prior. And usually, Risso's dolphins inhabit continental shelf waters. Puget Sound is not considered part of their normal range. So lastly here, some kind of management implications from what we saw. Gathering baseline data is extremely important for management. We did see high densities of harbor porpoise in Admiralty Inlet, which is an area that at least about 5 years ago was being considered for tidal energy production. And understanding things like, where the highest densities of certain marine mammals are, and understanding those group characteristics and behaviors helps inform related management in Puget Sound to avoid animal ship strikes, managing vessel traffic and speed, fishing seasons and areas to minimize entanglement, estimating numbers exposed to underwater noise produced by Navy sonar, and assessing impacts to endangered salmonids through marine mammal predation. So of course, we want to acknowledge the funding comes from the U.S. Navy and the U.S. National Marine Mammal Laboratory and the National Marine Fisheries Service. And so I do have to head out because I'm in the middle of a work day. But if there are any further questions towards the end, you can contact Mari Smultea, who's the corresponding first author for the paper, or Tom Jefferson, who's also on the call. Thank you.
Great. Thanks, Becca. Excellent work. I'm sure there'll be lots of questions for you, but we'll let you go. And yeah, feel free, folks, to activate that chat and send some questions along, and we'll make sure that they get to Becca. Cindy, are you interested in giving a 5-minute update for? I think I saw Cindy on the call. You may still be muted, Cindy. All right. I think she's there, but
I don't think you want me to unmute. My daughter is having a moment and I can't get her to stop crying. So I'm
no worries. We can come back to you after the next one if you want.
okay. I mean, yeah, I mean, maybe she'll, are you okay right now? You say hi to everybody. I can see you. There we go.
Yeah, I do see you.
Say hi. I mean, I can try. If she's distracted by you guys, she might be okay.
We'll keep waving. Yeah, if you've give it a shot if you want, but it's also totally fine to come back whenever you're ready.
Okay. Well, I don't know how long she's okay right now, so let's go ahead and try it and see. I don't have, it's not a huge update, so I won't be probably too long.
Great.
Okay, so let me, oh, is there a thing to share screen? There we go. I need that. Share. And if I can go, you see, look, you see? Say hi. Okay, that's fine.
Definitely seeing your screen.
Okay, so thank you. I'm going to speak a quick update on a paper that we're working on right now with our colleagues down in the Sound, Dave Anderson and Lori Schuster. I know. And no, I can't go for a walk. So we're looking at harbor seals in water large grouping behavior, which is not normally known for them to do. I know. Okay, I think we'll have to come back. I'm sorry.
That's okay.
This has been a day.
You can also, if you just email me your slide deck, Cindy, I can share it with folks. Yeah, or we can also easily push you onto the user agenda too.
Okay, I'll try and send you the slide deck just in case, and I'll let you know in the chat if she comes down.
No worries. Have a nice walk. Okay, let's see. I think we're a little over time, but I'm wondering if John Scordino, are you ready to give a little update on sea lions?
Yeah, I can do that. Can you guys hear me okay?
Yeah, you sound good.
Okay. Cindy, I think you'll have to turn off share screen so I can share mine. All right.
Starting to see it. And looking like full screen.
All right. You guys all ready? So I'm going to give you guys a quick update on some Steller sea lion and California sea lion work that we've conducted here off of Nia Bay over the past, I don't know, 10, 15 years. And so I'm going to present on behalf of my co-authors, Adrianne and Liz. Our study area is on the periphery of what you would consider on Puget Sound, Tatoosh Island being probably on the western edge. And a lot of the work that I'm presenting is going to be off into the Pacific Ocean side, because that's just how we research our area, is just all that area combined. And so you can see it's haul outs expanding the coast from Tatoosh Island down through Sea Lion Rock to the south. So first, let's talk about trends. And these trends are in the counts, the average count that we see within a year of the two sea lion species of Steller and California sea lions. they've both been increasing rapidly through our study time period, with a 7.8% increase for California sea lions and a 7.9% increase for Steller sea lions. And I should mention, I haven't updated this figure. This figure was included in a publication by Liz Allyn, sorry, and myself, and on entanglements of sea lions. And since this time, the Steller sea lions have been continuing to increase. And we've actually noted that Steller sea lions are now pupping in Washington. We've had the numbers of pups have essentially increased over the same time scale, that we went from almost no pups to well over 100, 150 pups a year right now. And then I should note that California sea lions have been lower in the last couple of years. So they have not continued that same trend, but it's something that we're going to continue monitoring, see if it jumps back up, or they were just feeding in a different area, or what was happening there. Second part of this, I want to talk about our diet studies on the sea lions. So we conducted studies of California and Steller sea lion diets in 2010 to 2013. We collected scats year round from January through December. Obviously, it's harder to collect scats in the winter, but we still had good representation of all seasons within the year. As you'll see here, our most common prey family groupings that were observed were your herring family, the Clupeids, rockfish, salmon, skate, dogfish, flatfish, and hake. And one of the goals of our analysis was to evaluate how much overlap there is in the diets of the sympatric species of sea lion, to see if there's competition among them. And they did have significant dietary niche overlap, but despite that, both populations have been increasing until just recently, where the counts of California sea lions have declined. But I'm not sure if that has anything to do with that competition or some other factor. Within our study, we decided to do a little bit more of a focus on salmon in their diet and the diet of the sea lions, because of their commercial, ecological, and cultural importance to tribes like Makah and other groups. What we found is that when sea lions are eating salmon, that most of the salmon they're eating are coho, and chum is the next most frequent species. We also found that Steller sea lions are more frequently consuming a medium size class to small size class of salmon, whereas the California sea lions are more likely eating a large size or to medium size of salmon. And I should say these small, medium, and large kind of break out as about a smolt, first year in the ocean size for small. A medium is that ocean growth phase where they grow anywhere between, say, about 12 inches and about 20 inches, and large is greater than 20 inches. And one of our goals really was looking at how much competition the sea lions might have with southern resident killer whales to utilize Chinook. And as you can see, it's actually a pretty small portion of their diet. And I'll show a little bit more on that in a moment. Now, to give our study perspective on what they're eating, I thought it was important to figure out how much of it they're eating. And I used published literature and some of our study results, including our count data, to come up with a prey consumption model. And so using the formula that's printed there on the screen, we developed this model to estimate how much prey California and Steller sea lions are eating per year. And just to note that this paper and the diet study has been accepted by Fisheries Bulletin, and hopefully will be published early in 2021. So here's our prey consumption estimates. As you can see, the sea lions are eating the most prey during the fall time period, and this coincides with when we have really high counts of California sea lions on the coast that are migrating through. And then we also have just a lot of more Stellers observed. And so with the increase in abundance, we're seeing an increase in their prey consumption estimates. So on an annual basis in that time period of our study, we are estimating that Steller sea lions eat 11,000 metric tons and California sea lions eat 9,000 metric tons. And so it's important to note that already, because of the population growth that I showed previously, these estimates are out of date. And with that 8% a year growth rate that I presented previously, these prey consumption estimates probably should be about doubled, if not a little bit greater than that at this time. So they're eating a lot of prey just in this area off the Northwest coast. And so a little bit more detail on our salmon consumption compared to commercial. I decided to put in the comparison to commercial fisheries. So first, you can look at on this table here that I have listed the metric tons of salmon that were consumed per year based on our estimates from our consumption model. And as you can see, our SSL here are Steller sea lions, CSL is California sea lions, SL is just sea lion grouped together. So if we look at our Chinook fisheries in coastal Washington, our commercial fisheries for Chinook are taking about 400 metric tons per year. And this is much more than what the sea lions are consuming, where they're consuming just about 200 metric tons per year with all of them together. And if we consider just the large salmon that the commercial fisheries are utilizing and are also being utilized by the southern resident killer whales, it's only about 47 metric tons that are consumed by the sea lions per year during our study period. Interestingly, it was coho that the sea lions were really utilizing frequently. And the prey consumption estimates for coho during that time period were actually pretty comparable to what the fisheries were catching. So if we look at Steller sea lions and all of their consumption of the coho salmon, it actually exceeds what was caught by fishermen in coastal Washington. And so that's comparing everywhere from Nia Bay south through Westport area for those commercial ports, how much they were catching. And sea lions were consuming more coho, as were California sea lions. And if we look at the entire state of Washington for the fisheries, the 1,600 metric tons that were caught in the commercial fisheries, which is pretty similar to the 1,460 metric tons that were consumed by all sea lions within Northwest Washington. However, I should point out that within the summer time period, the sea lions in our area make up only about 40% of the abundance estimate of sea lions in the state. So this is, a comparison that's it's not too direct comparison, because the state fisheries is the whole area, where we're just talking about a small area. We should also remember that the abundance of the sea lions has probably about doubled till now. And so these consumption estimates are probably also double. So now currently, our sea lions are probably consuming much more than our commercial fisheries for coho salmon, and are probably competitive with them on some of the other fisheries. Future work, just to keep you guys up to date with the other projects we're doing. We have a collaboration with Western Washington University and our marine ecology program here at Makah Fisheries. And so we're working on, again, collecting more Steller sea lion scat and harbor seal scat. We actually collected all this scat already, and we have a contract with Washington Department of Fish and Wildlife, and they've done metabarcoding analysis of the fecal samples, and they're doing prey identification of the hard parts. And all this work will be presented by Zoe Lewis in her thesis, and hopefully in a publication before or soon thereafter. And I also want to mention that we just received funding from Washington Sea Grant to do modeling, and this work will be done by Liz Allyn. And this will be evaluating how Native Americans resuming their harvest of pinnipeds would affect the rest of the ecosystem, and really focusing within that on salmon. And with that, I will conclude. So thank you, and I'll quit sharing screen here if I can find the button for it.
You're doing great. Super. Thank you very much, John. And let's take questions in the chat for John. And we're going to take a break now, but we're really pretty flexible in terms of the afternoon schedule. So I see, Holly, that you're actually present rather than traveling. So I wondered if you're ready to give your presentation, and then maybe we'll follow that with about a 10-minute break.
Yes, no, that's fine. Yeah, we ended up getting home a day early, so I'm going to present on behalf of John as well.
Super. Thanks for being live.
Okay, so I will share.
Starting to see it. We got your PowerPoint.
Yes, and can everybody see?
It's looking good. Full screen.
Okay. Okay, good. So today I'm going to focus on our talking about our southern resident photogrammetry work, but I can just briefly mention that going into 2022, we're going to continue our collaboration with John Kalamakidis and Cascadia on the Puget Sound Sounders gray whale. We've been doing some photogrammetry work and health assessment work of that local population. And we're also going to be continuing our BIGS photogrammetry work. So we've, over the years, we've been amassing a large data set, and we have a paper actually in review right now for the first size estimates and body condition based on our photogrammetry. So that's exciting from the big side of things. But going into 2022, we're going to be going into our 15th year of the health monitoring of southern resident killer whales. This is a collaborative effort between myself with the SR3 and John Durbin, who wears a number of different hats these days, but I've included his Oregon State affiliation here. And as I mentioned, it's a team effort. We started back in 2008 using helicopter, so the manned aircraft, and then we shifted into using unmanned aircraft or drones. This just shows John and I off of the San Juan Islands, and you can see Maximus, which is our primary research tool. It's an octocopter drone. And we relocated in 2019 to where we're based in the San Juan Islands full time. So because of that, we've really been able to increase the resolution of our monitoring, and I'll get into more details about that. But there's been a long history of concern with if the southern resident killer whales are getting enough food. I'd like to highlight that this is an article back from 2008, and this is actually what spurred our initial efforts to collect the aerial images so we could identify if the whales were indeed getting enough food. You could see this shows a graph from John Ford's work in 2009, and he showed an increased level of mortality that was linked to declines in Chinook salmon abundance. So you could see, especially in those years, the 1990s, where you saw the pronounced decline in Chinook abundance, and you had that increased southern resident mortality. But this problem hasn't gone away. I showed this is 2019 from Linda Mapes. You can see there's still a high level of concern on whether the southern resident killer whales are getting enough food. And as I mentioned, we're now going into the 15th year of our monitoring, but we're able to assess body condition, nutritional and reproductive status of southern residents at both the seasonal and an annual level to address these concerns. This just shows an image of us. We actually pioneered the use of drones to monitor the health of free-range cetaceans. Started that effort with northern resident killer whales back in 2014. And this actually shows a smaller hexacopter drone, but since then we've shifted, and this is another picture of one of the octocopter drones, to have a larger aircraft. You can see on the right, it shows the octocopter above the whales that allows us to very non-invasively research southern residents. So we fly at an altitude of greater than 100 feet. And because of the large size, we're able to stay in the air for a lot longer. We can get flights of over 30 minutes. And we're also able to position our research vessel several hundred yards away from the whales to minimize any kind of disturbance. And we collect amazing images like this. We definitely, it's ideal to get this beautiful clear water where you can see all the edges of the whales. You can see there's a nice group of the L55s, but from these images, we're able to do something called photogrammetry, and it's simply taking measurements from these images. And so this just shows you a couple of different examples of the kind of measurements that we take. But because we have a known altitude sensor and we have the focal length of the camera, we can get really, really precise estimates, less than a centimeter. The measurements are used to monitor growth as well as nutritional and reproductive status. And so I'll get into more details about that. And we're able to monitor individuals, which is really important. We're based on the distinct pigmentation you can see in the saddles from the aerial images. These are two examples of boat-based images. We're allowed to link all of our measurements to individuals of known age and sex, which is again really important to identify how each age and sex class is doing over time. And we're able to do all of this because of the work from our colleagues at the Center for Whale Research. And so we continue to collaborate with these guys. And from our work, and this dates back to the initial data that we collected using the helicopters, we're able to get size and age now for the entire southern resident killer whale population, but also based on our work for several years up in Telegraph Cove, we were able to get size and age estimates for northern resident killer whales. And across the two populations, we found a similar trend, and that these younger adult females, so you have the length in meters on the left axis and then age across the bottom. But you could see that these younger adult females aren't getting to be as big as these older adult females. So after they've stopped growing, they aren't reaching the same large size. And we suggested that this was due to nutritional stress. So if you think back to that John Ford graph, which showed that 1990s decline in Chinook availability, that this was a period that was really important for these whales to grow. So we found a similar trend between both the southern residents and the northern resident killer whales. And we just started up this past summer a study on Alaska residents. So we're going to be extending that comparative study to see what kind of trend we see in both size and condition compared to southern and northern resident killer whales. And one of the key metrics that we use to assess body condition, killer whales make it pretty easy in that they have these white eye patches. And so we developed something called the eye patch ratio. And it's very simply a proportion of two measurements on the head, on the eye patches. This just shows you three examples of varying condition of southern resident killer whales. And there's a northern resident killer whale on the far right here. But you could see that if they're healthy, you have that angling outwards of those eye patches. So you could see this is an individual that's still alive. In the center, you can see this is an image of J28 just a month before she died. So you could see that there's eye patches. They start angling inwards. They start tracing the shape of the skull. And on the right, you can see this is the northern resident killer whale. This was taken a week before she died. So you could see that those eye patches, they trace the shape of the skull. You could see that she's lost that fat along the body axis. So we really focused on this eye patch measure, but you could also see the differences in the profiles in the whales. We do take full profile measurements of everyone as well. And using this eye patch ratio, we're able to track the entire population over time. So this just shows you three images of three adult females, and then three graphs at the top. The eye patch ratio, you can see it ranges from under 1, it can exceed 1.3, but this just gives you an example. The gray lines show you the eye patch ratios of the entire population, and then the black lines that you see are for the individual image below. And so it just shows what kind of resolution we have. And this is for September measures, and I'll get into the seasonal measures next, but this just shows that we're able to track how these individuals change, if they increase in condition, if they decrease in condition, dating back to 2008 from the start of the study. And this is something that, again, we're continuing into 2022. But one thing that's really important, and it's one of the reasons why we relocated, or historically we'd had a dedicated effort in May and then in September, but Southern residents have definitely changed how they use the Salish Sea and their residency here. So we had a couple Mays where they didn't show up at all. And so we relocated, we're able to monitor them throughout the year now. And it's a really sensitive index because we are trying to identify if and when individuals change. And we can use this kind of information to help inform management groups. And you can see this is the body condition index. So this is the eye patch ratio again on the left. And it shows, for an example, with J36, how she changes from season to season. So this is based on the historical, the September to May. And we saw a trend that you had animals declining in condition as they moved into the spring, and then you had that increase in condition into the fall. This year, we've been able to actually image individuals in 7 months of the year, which is great. So before last year, we were able to get them in 5 months. So this is increasing to 7. So it's really providing this increased resolution for identifying what they look like in the winter, what they look like, throughout the year. We were able to just get L's for the first time in November, just recently, which is really exciting. So we're just building upon this seasonal monitoring each year. And across the year, we were able to get the entire population imaged, and we were able to get out every time they showed up in the area. But this just shows another example of the kind of fine scale resolution we have for tracking individuals over time. A number of the next slides are going to highlight the work done by a postdoc that was based at NOAA Southwest Fisheries Science Center, Josh Stewart. He had a manuscript that came out recently that highlighted or that showed the link between our body condition data for Southern residents and it linked it to survival and prey, which we're really excited to get that manuscript published. This just shows a single example, J17. And one of the things that we were able to do in collaboration with Josh is that we separate out each of the whales in the population into different categories. So you can see here, we separate them into calves, juveniles, subadult males, subadult females, and senescent females. And so we're able to have these categories, able to track the individuals over time, which you can see they're shown by the light blue and the light orange dots. But then just to show you right here with J17, and you could see the images below, that we're able to track these single individuals as well, and especially when you can see this notable decline up until when she died, 2019. But we're able to, based on these eye patch ratios, we're able to assign body condition classes to these individuals. And so you can see at 2015, when she was pregnant, she falls into this body class condition 5, which is the most robust condition. But then you could see as over time, 2016, 17, 18, until when she died, that you actually had this decline in body condition class, which is really important that now we're able to actually take these eye patch ratios and based on their age and based on their gender, we can see how they compare to the rest of the population. And one of the things that Josh showed in his paper is that this body class condition 1, so this very poor condition, the lowest condition, individuals had a 2 to 3 times increased mortality. So they were 2 to 3 times more likely to die once they fell into this body class condition 1. So this serves as an early warning system for us that we can take all of our eye patch ratios and our health metrics over time for Southern residents, and after each season and data sampling collection effort, that we could actually place these animals into specific categories and see how they're doing. On to this year, I'm sure a lot of you are familiar with J56. It's a current whale of concern. We actually noticed, even in 2020, it was a very thin whale. It was abnormally light in coloration. And then the last year's September health assessment that we provided, NOAA and other management groups, it was just on the borderline of that body condition class 1, but we did identify that it was a whale of concern, but it hadn't quite entered into that lowest condition class yet. But we continued to monitor J56 in the condition, and it did decline under this body class condition 1 this past September. And so we were able to identify that, collect those measures, and we were able to provide those to the Department of Fish and Wildlife. You could see here, this just shows, again, those different categories with the calf, the juvenile, subadult male, subadult female, adult male, adult female, and senescent female across the top, both split into J, K, and L pods. And the bars that you see, it's the distribution for the whole population across our entire time series. But you could see this black dot shows where J56 fell in, and that was from this year. And we're updating all of these measures now, and we'll have a report available by the end of January. And so we reported again this information to NOAA, a number of different colleagues, and to the Washington Department of Fish and Wildlife. And there was an emergency rule that was ordered asking for more space from the whale watching vessels. So this is the kind of real-time assessment that we want to be able to do to provide this important information to different management groups to try to ensure that the whales have increased access to the food when they need it, especially in vulnerable times like J56. And again, this is an early warning system. This just shows a graphic of the eye patch ratio and across time. But 8 of the 10 deaths since 2015, for the time period 2015 to 2019, were preceded by a decline in condition. So we can detect these declines in condition. We can detect when they fall into this very poor condition class. And we hope that it can be an early warning system before whales die, so we can identify these animals of concern, and some actions might be able to be made to ensure both increased access and potentially increased availability to their primary prey. We're able to track these individuals at a population level as well. This again is from a report that to the NOAA West Coast region. The condition index here is actually the proportion of individuals in non-poor condition. You can see condition index is shown with the black lines, and then the abundance is shown with the red. But you can see there's a correlation between the two. So it's split into J, K, and L pod. And one thing to note is that when you see these declines in condition, they precede these declines in abundance. Same thing with the increases in condition and increases in abundance. So it's a very important metric for predicting what might happen with population level. From Josh's paper too, he was able to link the condition to different prey availability and prey stocks. So you could see on the left here that you have the probability of growth, the probability of moving into that increased body class condition level. And you could see that it was highly correlated with the Fraser River Chinook for J-pod. And with the L-pod, we found a strong correlation, not as strong with the Fraser River and J-pod, but with the Puget Sound Chinook. We didn't find a strong correlation between K-pod, but again, one of the things that's confounded a lot of the work with southern residents and different prey availability is how highly mobile they are. And so it's definitely something that we're adding that seasonal component of our monitoring in as well to try to target that. And again, it involves both abundance and accessibility to their primary prey. And it's something that I know a number of different groups are working on, is what are the best next steps forward to try to ensure that they get the prey that they need. Again, we're working with Department of Fisheries and Oceans in Canada, with Washington Department of Fish and Wildlife, and with NOAA, especially West Coast region, that we're providing these health metrics to them after each effort and also at the end of each year. In addition to monitoring this body condition and this nutritional status, we're also able to identify when individuals are pregnant and monitoring this pregnancy. So the success, hopefully, but definitely not always. This just shows you with killer whales, they get big just like humans do in the middle. You can see this is an image J35 taken September 2019. I drew the red line there. You could see July 2020, how much bigger she gets in the middle. And then September 2020, you could see how big she was. They're awkwardly large, but this is right 17 and a half months pregnant, right before she gave birth. And you could hear this is a positive end with J35, with the new J57. But unfortunately, with southern resident killer whales, you have a really high level of reproductive failure. And Sam Wasser and the Giles and the University of Washington group documented, more than a 68% failure of reproduction. With southern resident killer whales, we found similar results with our body condition data. So this is something that, and this next year, we're working on getting several publications out looking at the fecundity and relating that to body condition of southern resident killer whales. But again, this year, working with Washington Department of Fish and Wildlife and NOAA, and the similar actions were taken and asking for more space last year for identifying pregnant whales. We were able to identify 3 pregnant whales, and again, they're considered to be vulnerable. There's an emergency rule that was issued with J36, J37, and J19. There are also a number of other whales in the population that are pregnant. So far, there haven't been any new calves, but we're still hopeful. We've been able to see some recently, so still hopeful that we'll have some new little ones in the population. And again, this brings to light just the need for real-time metrics. After a single September effort, I just was going through all of the images. We had almost 30,000 images. That's just for one single effort in September. So it's definitely, we're trying to get through all of the analysis as quickly as we can. And Scott asked me to just touch base on a collaboration that we have with Vulcan, which is now actually shifted to the Paul Allen Institute of Artificial Intelligence, so AI2. They had a bit of a shift in their conservation program. But we've been working with them coming up now on 2 years on trying to develop an automated program or tool that we could use to try to speed up our southern resident photogrammetry analysis. We've had some progress. We're going into phase 2 now. So we have a tool that we have been using, which is great, but we're going into phase 2 as far as the automated development of the project. So we'll see how that goes, but it's definitely been a real pleasure working with them, but it's an ongoing collaboration. And I'll just end with that. And again, sorry, John couldn't join, but.
No worries. Thank you, Holly. That's exciting work. And I've linked to a blog post that I was excited about earlier in the summer. So if folks want to explore the AI side of things, maybe we can revisit that in the winter when I think we'll come back to talking about body condition and ancillary measures related to the vital signs. But in the spirit of COVID and adaptability, I want to keep pushing our breakout just a tiny bit more. Stephanie is doing a little dance with an elephant seal this afternoon, and I want to give her a chance to give her a presentation about belugas before she may need to run off in about 15 minutes to attend to an elephant seal. So if you're ready, Stephanie.
Yeah, thank you. I'm sorry about this. It's like coming and going. So I found 15 minutes here to give a talk. So do you want to share on your end? That would be great. And then I can just tell you when to.
I've got it queued up here.
So these slides, some of these slides are from the talk I gave at the beginning of November that Scott alluded to. That was for the Pulse program. And I think Cindy Hansen's on here. So anyway, this was just talking about, in the initial talk, we talked about, belugas in general. And then we talked about, the Cook Inlet belugas, because that was one of the stocks that is endangered under the ESA. And so it was one of the stocks that there was of concern as far as whether this animal was a match for that. But I'll go ahead and, okay, go ahead. Yep. So this is the video that started it all on, let's see if we can play that. It's just a short, like 20-second clip. Oh, maybe not.
Yeah, I might have lost it when I brought it into Google Slides.
Yeah. Okay, anyway, it's just the initial clip that Howie and Susan received the notification back on the beginning of October. It was on a Sunday afternoon. And then the location here, the red dot, is where that video was taken. And it's funny because in the video, you can hear someone say, that can't be a beluga. And, I would be saying the same thing down here in the Northwest. But so this is location, Commencement Bay, where that initial sighting took place. And then go ahead, Scott. So just kind of go back, first we started thinking, gosh, have they ever been seen here before? And Scott nicely put this together on the Google Drive that we had set up for this animal. Going back, there was a sighting back in 1940, and I think it was down there for about 3 to 4 months. It is mentioned in collection of sighting information by Sheffer and Slip in 1948. And there was even a Tacoma. Linda Mate from the Seattle Times said there was in the April 1940 edition of the Tacoma Times carried a story about this, kind of unknown, weird looking white mammal that was seen by locals. And the funny thing here, it said it was headed to Olympia to speak to the governor. So I thought that was kind of funny. And then there was, and I don't know about this one, you might be able to chime in, Scott. There was a potential sighting in Saratoga Passage in 2010 or a reliable report of a beluga, but there was no photo. Do you know anything more about that?
I would defer to either Howard or Susan on that one, because
Yeah.
I don't.
Okay.
Do it.
Okay. Well, we can.
Yeah. There,
Susan?
yeah, the last report we got was off of Newport.
Oh, I was talking about the 2011 Saratoga
Oh.
Passage one. Do you know anything about that little sighting?
Oh, I'm sorry. Yeah, that was, it was Florian Groner, a photographer, but it was his father-in-law. Florian was diving, I believe, and his father-in-law swears he saw, and what Florian said described clearly what sounded like a beluga, and really he couldn't think of anything else it could be, but there were no other sightings. So it's kind of a second-hand, non-confirmed, no photo report.
Yeah. So, you know, possibility, and these are sort of the sightings in pre-2021. Now, there was the animal down in San Diego that was sighted a year ago, 2 years ago, but then they think eventually it was the animal that washed up dead in Mexico. And unfortunately, they weren't able to get a hold of that specimen to necropsy. It was sort of a third-hand report from a fisherman. Okay, Scott, go ahead. So Scott did a great job of summarizing. He had a spreadsheet of all the sightings. Oh, thanks, Tom. I see that. Yeah, it was in San Diego in June 2020, the other beluga. So this is just a kind of a list. Every time someone saw the animal, you know, it was reported, it was logged in, whether it was through the sighting network or, you know, just someone calling in one of the stranding groups or directly to anybody and trying to get these, you know, confirmed. And then where they are not confirmed, there's a little, you know, a note is made. But it was, you know, people were getting used to seeing it. There was some good footage, you know, pictures that were taken. So people were becoming familiar with what a beluga looks like. So the public was very helpful in this and provided us quite a number of, you know, sighting. And go ahead, Scott. So on the 7th, so this is 4 days after the initial sighting, Scott and Michael Doherty and Diana and Greg Frankfurter, myself and Kathy King from World Vets and Amy Van Sice from the Northwest Fisheries Science Center, we converged down in Tacoma because that was the last time it had been seen down in Commencement Bay. And we proceeded to do a field assessment to just try to get some information, all these different parameters, photo ID, to see if we could match it. There is a photo ID project that has been ongoing for several years in Cook Inlet, and I think a little bit in Bristol Bay. It is, you know, a challenge to try to match up these guys because they, you know, they're all white and they don't really have these large distinct dorsal fins, but they've done a really good job of being able to identify individuals. I don't know how many off the top of my head, but there's quite a number of them. The last known count, I think, on these guys is like around 372 for Cook Inlet. So we did, so there are three of us vets down there. The animal was determined to be a little bit thin, but not necessarily emaciated. So it was at least hopefully finding food. The behavior on this guy, erratic, you know, it was all over the place. We kept, you know, trying to go from, it was sort of like whack-a-mole, going from one place to the other. Not necessarily that it's abnormal behavior, it's just that, you know, in certain parts, like in Bristol Bay, they tend to go to the same locations. In Cook Inlet, same thing. So you can kind of reliably, if they move here, they're going to go here. But with this guy just being one animal and trying to track it in a big body of water, there was a lot of ship activity going on. And it seemed to sort of, our impression that it was sort of drawn to these busy areas. One of the goals was to, again, conduct sampling, eDNA. So Amy Van Sice came down with the equipment to do that from Kim Parsons' lab at Northwest Fisheries Science Center to try to get a better handle on, you know, can we get some biological samples from this guy, whether it be fecal samples, skin, slough skin, what have you, to try to determine some approximation of the stock that it comes from. And also acoustics. So Scott and Michael, you know, and also Kathy put in hydrophones, and Scott, you can talk more about that and how, if that was the challenges of that.
Yeah, basically there were so many parked cargo ships in that Tacoma area that the ambient noise levels, which should have been like 105 decibels, were more like 117 everywhere. So it was kind of hopeless from a signal to noise perspective.
Okay, so going down, next slide. So Scott put together these great slides, that these maps of the, so this is the first week, you know, really busy sightings. Now, I'm not familiar with the path that it necessarily, wasn't there supposedly a possible sighting up in Canada before all
Yeah,
this started? So maybe you can speak to that.
Yeah, it was the Gulf Islands one, and it wasn't photo confirmed, but it was pretty compelling. So I added it in as a possible route in from off the Cowichan River Delta.
Yeah. And so this, this animal would, hang out in Seattle, then it went across to Bremerton around the busy naval shipyard there, and then it would come on down to Tacoma as far, keep going further south into Lakewood. So it was, it went around probably following the food, or I don't know, maybe getting its orientation of where it is and where the grocery stores are around there. Okay, Scott, next slide. And second week, again, it was coming all the way down, back up to Everett area, off of Whidbey Island, and then continued down through the South Sound, down around Vashon, and then all the way back up to Port Townsend area. And then, I don't know, I know Diana's on here. You might have some more information on any sighting information that you had, so feel free to hop on, or we can do it after I'm done. Okay, next slide. So the last confirmed sighting was for the 14th of October off Lagoon Point in the east part, Whidbey Island. Is that off the East Channel, Whidbey Island?
Off of Admiralty Inlet.
Admiralty Inlet, okay. Now, okay, this slide's old because I forgot to send you the updated. So before the 13th of November, the last unconfirmed sighting was the 20th of October near Tacoma, but this should instead read the 13th of November off of Newport, Oregon. So that was the unconfirmed sighting. We did, during this whole period, there's a lot of stuff going on in the background, a lot of activity, because there was some thought that this might be one of the endangered Cook Inlet animals. So there were contingency plans in place in the event whether it was a live animal that's stranded or whether it's stranded dead. So Kristen Wilkinson and others were in contact with, beluga experts up in Alaska and other places about, what to do if this animal strands, particularly if it's a Cook Inlet animal, because it is a high priority animal. And that was sort of the impetus to try to figure out with the eDNA what it is, what stock it came from. And then also if it's stranded dead, we got information on particular samples that they wanted or of interest on this particular animal. So the publicity, it's a funny thing, the ironic thing is on the 7th when we were out there, it was really a challenge to find this animal. And all of a sudden, we got information on this location from the Como News, or King 5 News was doing a live feed of this animal swimming around. So at least we were able to get over there. The irony of the media on top of it, but it did help us in the end get a little bit more one-on-one time with this animal to try to get photos, acoustics, and some eDNA, which we were able to do. So I ran that up to Kim that night at the lab. The good news is it wasn't a match for that we're able to get some samples from out of the water sample, and that was not a match for the endangered Cook Inlet. So it did kind of take sort of the pressure off, like, oh my gosh, this is one of those animals. More likely it's an Arctic or Beaufort Sea animal from one of those stocks. But unfortunately would need to have more information such as a skin or fecal sample to more definitively ID the stock. And I just have a little map there to show you where, and then Scott has a slide on the next slide, but just kind of where the stocks are recognized in Alaska and where they are situated. So on the next slide that Scott has, basically this guy, super traveler. Belugas are not like, gray whales. They aren't necessarily going to go, thousands and thousands of miles, but who knows what's going on, unknown what could be going on with this animal, if it's food related, if it's, disease related, health related, we don't know. So at this point, it'll be interesting to see if there are any more sightings of it, if it works its way down the coast. And who knows, the one in San Diego might have been up here. We just never knew it. We never detected it. So I think that's all I have on this. But yeah, it was a neat case, and we'll see what happens if we see it. And so now we're adding this species to the list of the Encyclopedia of Puget Sound. So we're in the process of writing that up. And any questions or comments?
I just wanted to thank, and I think Elisa Lemire Brooks, our sightings coordinator, is on the meeting, unless she's back out with JPOD. But I just want to, if she has any updates on other reports, but I also want to just thank her for all her hard work, working with Kristen and with the other researchers, and really being on the ball, keeping everyone updated on the sightings for this whale.
And we did give a heads up to California, and I had mentioned to Kristen to also alert the Mexican stranding networks that in case it does go that far south, if they hear about a beluga, then it's probably not a false report. It could be actually a beluga. So hopefully if it is down there, we'll see it again and be able to follow it up.
Yeah, I let Steven Swartz know also the other day, so he'll help spread the word.
Well, super, Stephanie. Thank you for
All right, I'm off to go check on an elephant seal.
Yeah,
Thanks.
good luck out there. Super. Let's take a 10-minute break, and then we'll come back at 3 o'clock and hear from Giles. And I think Cindy may come back once more, right? We'll be back in 10 minutes. Okay. Thank you. Clockwork, well-oiled machine here. All right, so it's 3 o'clock. We've got an hour left in our agenda. So let me just share my screen briefly as folks are coming back. So I think next up, we should go to Giles and Cindy, and then I'd like to just give a brief update about the Orcasound hydrophone network. Then we can segue into updates, just quickly run through all of our species and make sure we're not missing anything in terms of 2021 observations. Giles, are you back and ready to give a little update?
Hi, everybody.
Hey, welcome.
Yeah, I just got my booster today, and I'm so flushed and feeling super warm. So I'm glad I'm going now, and I think I'll just lounge around on the couch and listen to the rest of the meeting after this. So yes, I'm ready. Great.
And I'll take it away. And ideally, we keep these to like 5 minutes each, but it's okay if we need to flex. That's all right.
I think it'll be faster than that.
Great.
Just wanted to make a quick announcement, and I'm trying to say share here. It says I can't share while others are participating and sharing.
My bad, that was me. Let's give it another try. As your temperature rises.
Yeah, it's so crazy, just suddenly. But I'm taking that as a good thing. Okay, and can you see that?
That looks good. Nice pup.
Okay, yeah. So I'm really excited to announce a couple of people know this already, and it's been out on social media a little bit, but we haven't talked about it too much. But Sam Wasser, Dr. Wasser, is passing the research, the Orcascat project, on to me and Wild Orca. Very excited about that. I've been a part of the program since 2009. He started it in 2007. But I've been part of it for a long time now, and excited about expanding it in a way that I feel it's needed to be expanded. Just as a quick aside, Sam is doing some phenomenal work. He's started a new program, a new department at the University of Washington in partnership with about 40 other researchers and organizations from Interpol, Homeland Security, any large organization that has anything to do with dealing with illegally harvested and trafficked endangered species. And so it's going to be called the Center for Environmental Forensic Science, CEFS for short, and it is a new program or new department at the University of Washington. So the Center for Conservation Biology has now sunsetted, and then his new department is CEFS. So when that happened, a larger focus of SAM is going to be on genetics. and being able to track the genetics of these species across the world. And so when that happened, it really became clear that the whale program needed a new home. And as I've been a part of it since 2009, it just made good sense to transfer it to Wild Orca. So we'll continue to use Iba, the whale dog. Anybody that's not following her already, you can follow her on Instagram. I hope to have some interns that are better at social media to help me out with this moving forward, so that we can very much keep up to date on this. We have a new concept about how we want this science to be done and made available as quickly as possible. And so what we're calling, so I just wanted to give a shout out to this amazing boat. Moja is now retired. We're in the market for a new used boat. Moja, I put over 3,800 hours on this boat, collected over 750 samples on this boat. And I will miss this boat very much, but we need a boat that is able to get us farther and faster in order to get us to the areas where the whales are spending more time. And so in the past, we've been really focusing our attention in and around the San Juan Islands. When we can get notice about the whales being up near the Fraser River early enough in the day, we could get up there in Moja, but it was just not very fast boat. And so our new boat will have the capacity to get us farther faster so that we can get where the whales are. And so our proposed expanded area will be out here to Swiftsure Bank and in through the Strait of Juan de Fuca, down into South Sound, where it's just been crazy making, knowing that they're down there as much as they have been. Brad has been able to get out a couple times to try and get fecal samples and prey remains, but not as much as we hope to when we have our increased capacity. So our concept here is moving, we're calling it from detection to protection. And this is kind of a schematic of how we envision being able to utilize these samples. So we want to be able to collect them, continue to do it non-invasively using our scat detection dog, process the samples much more quickly. So our lab is going to move from University of Washington to the San Diego Wildlife Alliance, and our partnership with them will allow us to have these samples analyzed much more real time. We're hoping for at the long time frame would be quarterly, but if we have a month where we have a lot of samples, we'll be able to do them much more quickly, like on a monthly basis, or even faster if we need to. So these are the Wildlife Alliance is the organization that is famous for their white rhino work, and probably most famously the California condor work. So they're experts in looking at reproductive success, and specifically in tandem with environmental man-made toxicants. So very excited about that, kind of moving from an academic pace to more of an actionable pace. And then being able to put those findings in the hands of policymakers as fast as possible. One of the things that was really, I thought, amazing, Holly touched on it a little bit, but what the drone folks are able to do is give these very much real-time feedback to management agencies. And I thought that was really fantastic for those pregnant females. And we'd like to be able to help participate in that real-time more real-time analysis of what's happening on the individual and population level going forward. So this is not new, this is very much what we've already been doing with the Center for Conservation Biology. Again, just being able to process these samples more quickly and get them into the hands of policymakers as soon as possible. So currently working on a Rose Foundation grant. Thank you so much, Rose. Bailed this out in a pinch last year, hoping for funding again from them again this coming year. And the Paul G. Allen Family Foundation has been a very strong supporter of this research, and hope to have that continuing going forward. So that's all I have. I'm happy to answer any questions. Otherwise, I do intend on sticking around, and we can, anybody having questions, we can come back to them at the end if you want to move on at this point.
Great, thanks, Giles. Yeah, why don't we had some success earlier using the chat feature, so keep an eye on that while you lie down and rest a little bit.
Okay.
So yeah, everyone feel free to hit the little chat bubble at the bottom of your screen and add questions or respond to them. But let's go on to Cindy and see if you let me know if you want me to present your slides or you'd like to share.
No, I'll put them up here because I can have the video, hopefully. My son is distracting my daughter, so I appreciate everybody's patience. It's been it's rough sometimes.
With you, it's a Monday for sure.
Yeah. Let's see, so share screen and we'll go here and there. Okay, and from current slide. Okay, does that look good?
Yeah, we're seeing it.
Okay,
Full screen.
awesome.
It's great.
Okay, so this we're working on this short note for the commentary reflection section of behavior. They put out a request for these type of opportunistic and observational things and interesting information. So what we're looking at is harbor seals congregating in water in large groups. So usually, as you've already talked a lot about harbor seals already. So, but usually you'll see them in larger aggregations on beaches and rocks during rest or pumping seasons. But generally, they're thought to be relatively solitary at sea. I mean, you don't normally see more than a few, maybe together, out in more open waters. Occasionally, there have been documentation of them in larger groups in the water, but that's usually associated with something specific. So swimming near haul out, so they're all there because they're being hauled out, or forced bottlenecking at channels or rivers where the prey items are and there's just limited space, so they're there because there's food. But open water, less that we've seen with these larger groupings. So our research, Pacific Mammal Research, we look at harbor porpoises and harbor seals, doing photo ID, long-term studies on both those animals. It's up at Washington Park off Fidalgo Island. So we've been out there since 2014, so about 7 years now. And with harbor seals, they're there year-round. We see them in small groups occasionally in the past. But usually they're only 1 or 2 at a time, maybe 3 that are kind of loosely associated. And we see them foraging there all the time. We see lots of foraging. We see mating displays, tail slaps, and actually we documented a mating actually right below us on the rocks, or in the water on the rocks that we sit on. So we have not seen until recently these quite large groupings happening in the water. So what we've found, and we're working with our colleagues down in the South Sound as well, which I'll mention in just a minute, Dave Anderson and Lori Schuster, who have also observed this behavior only a couple times, but still there. So we have about 32 cases that we've documented, these larger groupings, from 6 to 50 individuals with a mean of about 16.8. And the behavior is usually they're floating at the surface, as you can see in the picture, usually within two body lengths of one another. So they are together. They're not just loosely, just in the area. So we've seen 30 of these in Burroughs Pass. And like I said, this is a new behavior. We were out year round since 2014, and we have not seen this behavior until 2019. And we've seen it every single year since. And what's very interesting is that it happens in very late April, like 2 or 3 days of late April, and basically May, and then all of May. And then we had 1 sighting in June of this year. But it's very timed. It's the only time we see it is during basically May and that little bit of late April. Dave and Lori down in the South Sound have seen it in Carr Inlet and Case Inlet 1 time each in February 2017 and January of 2019. And they did recently see one this summer, which we'll be adding to the paper. And these are all, it's important to note that these are all away from haul out sites. So they're not congregating there because they're about to haul out or associated with that. These are more than at least a mile away from any haul out site. So this is another picture of how they're kind of spaced apart from each other, but all together in this group. And in our area, it does seem that they float down the pass. And then as they're floating, they will do this behaviors where they do inverted diving, periodically diving 1 by 1. So they'll kind of go inverted, as like they're fishing, kind of one at a time. Then a few of them will be down, then they'll pop back up. Then a couple others will dive back down, and they'll pop back up, and they're all floating together in that group. It's predominantly done during flood and slack high time. There were only 5 out of the 32 that were during ever slack low. So it does seem associated with tidal influence. And so, we're trying to decide, or not decide, but figure out what they're doing, why they're doing it. Like most of the cases, it does look like it's a foraging behavior. It's not rafting like other otariids do. We have not seen any mating behaviors during this time, so we don't think it's socially related. Like I said, we do see mating in Burroughs Pass and the tail slap displays and things like that, and that was never seen with these guys. And until recently, the predators, at least in Burroughs Pass, aren't really there. We don't see orcas there that often until this year, right? This year we've seen orcas go through there in July and August. It was crazy. They were there, once or twice a week, it seemed. So they were there pretty predominantly in comparison to previous years. But and that happened after June of this year, which is the last time we saw it here. So for the 3 years we've documented it, it's not really thought that this would be a grouping behavior because of predators going after them. So we do think it is a foraging one, whether it's linked to some new prey items or timing of prey items that happens during May, basically. We're not sure. We would love to look into that more. But we're just documenting the fact that we're seeing it now, and it's very interesting. Here's another picture. Again, you can see them inverted, and they're swim just underneath the surface, and before they dive down a little bit more. And then the last thing I have just to finish up is a video, which hopefully it will play. And I think this might have audio on it, so please disregard it because it's us just talking in the background as we're watching it. I haven't had time to edit it out.
We're seeing the video, but no audio, just so you know.
Okay. Yeah, I think I picked one that didn't have audio. So that's a pretty good video of what they will be just periodically doing as they're in this group floating along. And so we just thought that was very interesting. So we're going to be putting it out there, and that's all I have. And again, thank you guys for being so patient and allowing me to tack that back on in the end here.
Super, Cindy. Thank you very much. And maybe folks can jump in on the chat and let us know if you've seen that behavior. I have not. Totally novel.
Yeah, when we first saw it, I was like, what are you guys doing? You're not supposed to do that.
Yeah. Is there a link to a paper, or did you say that's in progress?
No, it's in progress. We're about to submit it.
All right. Super. Well, thank you for the updates, Cindy. I'm glad you were able to join us and make it happen. I'd like to share with you just a few updates from the Orcasound hydrophone network, which I've been helping coordinate for the past few years. Linked to a talk I gave about a week ago at the Acoustical Society of America meeting about the network and the emerging software solutions we're trying to share with other bioacousticians. But let me just switch to a different screen here. As just a quick synopsis, and I'll try to keep this under 5 minutes. Everything above this gray dotted line we've built over the last few years. We're trying to come up with a cost-effective way of listening in real time for for southern resident killer whales, but I'm hoping that everything we build for southern residents will be useful for other soniferous marine mammals, including eventually harbor porpoises. So, we basically have a flow of data from 3 locations where hydrophones are displayed through little single board computers that cost between $15 and $100, and an acquisition board called a PSEMP made in Lithuania, which is about $150. And it's not good enough to listen for harbor porpoises, but such things exist. And so we're trying to be really open about the hardware solution that we're building, as well as the software. But ultimately, the main thing that we've built in terms of new software is the code that runs on the little computer to generate these live streams of audio data, and the code that runs in your web browser on any device and any operating system that makes it easy to listen to the real-time streams. So if you haven't seen it, Orcasound, live.orcasound.net is where you can access the listening app. And it's an app in the sense that it's not just a website, it's a dynamic thing that changes over time. Like right now it's showing you that there's 6 people listening to the Bush Point hydrophone. And you can switch to other locations like Port Townsend or Orcasound Lab up in Harrow Strait. The goal, of course, is mostly to teach people what killer whales sound like and then get them listening. So the previous version we made so that when you're listening, you can also indicate when you hear something. So you can tag the audio data using this little button here. So that's been working really well, and I'm excited to share that, even it's basically becoming a real-time tool where humans are annotating in real time and machines are listening at the same time. So for example, these are all the button pushes by humans in the past few days. And you can see that last night at 12:45 in the morning, somebody was listening and said they heard clicks and faint calls at Port Townsend. And about shortly thereafter, nobody had reported anything at Bush Point, maybe because they had fallen asleep, whoever was listening there. But the machines that Microsoft volunteers have hacked together for us over the past couple of summers were listening and awake and detected southern resident killer whale calls. And for example, in this 60-second clip from last night, you'll be able to hear MS1 calls. And so each of these candidates is something that we've been testing, with Dave Bain and Veirs and I getting notified in real time when the machines hear things, and then moderating those, so going and reviewing them to make sure the machine was right, and then being able to review those and then issue a notification in a more automated way than we have in the past. So this system continues to evolve, and I just wanted to flag that, for a long time, the last couple of years, the human detection button and the machine learning side of this were red lines because they hadn't been fully implemented. But it's working, and I just wanted to say that in combination with the sort of amazing growth of real-time opportunistic observations through the sighting networks around killer whale habitat, we really are getting almost 24/7 ability to monitor location data. So the door is opening to aggregate acoustic detections with visual detections, and to use, to put better hydrophones in, calibrated hydrophones, and go back to one of the things we did at the beginning, which was measuring noise levels in addition to listening for whale signals. That's just a quick update, and I'll talk some more about that aggregation of spatial and acoustic data, or visual and acoustic data, in the Salish Sea Ecosystem Conference, I hope. So you feel free to explore my slides, and happy to take questions on any of that sort of human AI system that's emerging. We have about a half hour left, and my favorite part of these fall meetings is making sure that we all get a chance to share everything we've seen in the last year, and then over the winter, we can think about it some more and hopefully generate some new data products, some summaries of what's happened in the previous year by next spring. So with that in mind, I thought it'd be an interesting experiment to go through each of the species we monitor and make sure we're sharing, everybody has a chance to share unusual observations or patterns that our neural networks are detecting in the last year or so. So why don't we start with southern residents? Does anybody have any thoughts they want to add, either just by unmuting yourself, or feel free to add things in the chat too, but any new observations that we want to share about southern resident killer whales in the last year or so?
I'm hoping, is Elisa still on? I know she's also watching out with JPod right now, but she may have something.
I'm Maya. I do have one thing. One thing I noticed about the southern residents this year is that it seems like everybody has broken out with a case of whale pox. So I know that some people are kind of looking at that. I've never seen it this bad or affecting everybody before, but it seems like it might be one of those things where it just spreads through the population, then goes away. But if you're out with them, please keep an eye out for it.
Yeah, I can comment on that briefly. We haven't noticed anything like that from our aerial images. There was a lot of kind of skin sloughing almost with a number of whales when they first came into the area, but we haven't noticed anything that looks like pox. So just to add that.
Oh, sure. Do you want me to send you some photos so that you know what I'm talking about? And I assume it's whale pox. It might just be skin sloughing, but it might be nice to send some photos so you can kind of take a look at what we're seeing on the ground level.
Sure. No, that would be great. Yeah,
Okay.
I guess, do you have my email or access to it through the list?
Let's see. I'm pretty sure I can get it through the list here. Let me double check. Why don't you just put it in the chat and I'll write it down.
Okay, sure. No problem.
Mm-hmm.
Is that something you see all over their bodies, Maya, or in particular parts?
Well, you know, we're down at ground level, so we can really only see the top of them typically, but we're kind of just seeing it all over their front and back and dorsal fin, to various degrees and in various spots.
Great. I wanted to flag just a few patterns I've been noticing in the movement of the southern residents. And these may not be anomalous, but I think because the sighting networks and the both down here in Puget Sound near Seattle, but also up in the San Juans and the Gulf Islands are getting better and denser, that we're starting to resolve from land-based observations movements that I think are interesting, and combined with the acoustic detections. I just wanted to flag that it feels like there were more extended excursions up towards the Fraser, and we don't have great, the sighting densities tend to go down when they go north of the border. So I don't know where they went, but I've been wondering, thinking back to Brad Hansen's satellite tags that showed excursions up to and even, I think, around Texada Island. But it feels like they're paying more attention to that, the Strait of Georgia, spending a bit more time up there in the late summer and early fall. So anyways, I just wondered if anybody else had noticed that. And for me, in anticipation of trying to think more carefully about with the salmonid work group, I'm starting to think about other runs that at a more granular level, salmon runs beyond just, oh, the Fraser, summer Fraser run, like trying to think about what they're after at a more specific geographic run and runs within rivers. So anyways, just thinking about the chum and the coho runs on the Fraser, and then the runs on the to some of the rivers on Vancouver Island as well. And I guess in the same vein, I'll just quickly say that I'm not sure, but to me, it feels like this fall, there's been a remarkable, like the proportion of time the Southern residents spend around the possession triangle, which feels like it's higher than in past falls, the last 2 falls. And, they continue to not go south of the Tacoma Narrows Bridge, but they make excursions down to Tacoma. So I'm hoping today, they almost got to the Vashon hydrophone. I forgot to mention that in 2022, we've had Sound Action and Orca Conservancy join Orcasound as a part of the cooperative network. And so I think we'll have 2 more hydrophones, which will improve our ability to monitor movement on foggy days or at night in central Puget Sound. But I have been wondering about, where do the marine distributions of salmon lie in the fall? And I hope that's something we can think about with the Salmonid work group, because they're shifting from a general salmon indicator in a vital sign sense to looking at particular species of salmon. And so I think a door is opening for us to have a better integrated conversation with that work group about what the Southern residents may be after at any given part of their seasonal movements. But yeah, I'm wondering whether salmon, maybe it's a good foraging strategy to stay near Possession Sound because there's incoming fish to the Skagit River as well as all the Puget Sound rivers. If you go south of there, you may miss the Skagit ones and vice versa. So anyways, I just wanted to flag those as curious. Maybe when Elisa, it frees up, we can talk about the year-to-year patterns we see in the fall movement behavior.
Yeah. Are you there, Elisa?
Yeah, I don't have specifics. I wasn't prepared for that, but definitely in the last, including this year, last 2 to 3 years, they're spending a lot more time hanging out, foraging up in northern Puget Sound basin there off the Possession Bar. So just south would be there. So oftentimes I think we lose sightings. It's difficult for people to see them from land there. But yeah, definitely I agree with you that it's been kind of a hot spot the last 2 to 3 years. And the other change that we all know about is that they're very consistently spread out coming down the Sound, a mile in distance and across the Sound. It's rare to see them grouped up. Was lucky enough to see, I think, K-Pod a few weeks ago. When they were down, that was really lovely. When they were coming back down Possession Sound. And just, yeah, the shift in travel patterns, not circumnavigating. I think just as equally they turned back somewhere along the main channel of Vashon versus circumnavigating like they used to, years back.
Yeah.
So.
And it seems like they haven't been in Saratoga Passage as many times. I'm not sure if that's or out in the data, and we are working to get a hydrophone off Langley on the Saratoga Passage side and or in Possession, try to detect if they head that way. And I just have to give a shout out to Elisa and Gail, our team. We just got the November spreadsheet and we've collected over 7,000 orca reports so far for 2021. It's just due to the bigs coming in so much, it's been bonkers. They're working hard.
Yeah, somewhat related to that. I mean, I wanted to keep flagging that it's by far the best use of Facebook that I've seen. Public or private groups seem to really be growing rapidly, and I think it's an important and interesting new source of opportunistic observations. And it's totally resolved movements of big killer whales in ways that I just don't remember at all. So maybe we could move on to that. And while we have your attention, Elisa, I'd be curious to know if you or anybody else has, I know Monica's not on and she'll be able to join us in the winter when we talk a bit more about Biggs occupancy, but any thoughts on Biggs killer whales observations in 2021 that we should take note of?
Well, I think based on her putting data together, OBI putting data together, that, yeah, the numbers are up each year, the last several years for sure, for occurrences and duration of stays. And so right now, as far, Hood Canal came up earlier, but yeah, the T68Cs have been in Hood Canal for going on a month now. Haven't had sightings the last 2 days, so they may have exited, but they came in on the 19th, November 19th, with a larger family, the 68s and a couple others. I think 124, no, 144s. 164s, I forget. Anyhow, sorry. But 68 Cs tucked into Hood Canal on the 19th into the 20th and have stayed. So it's another prolonged stay for one family.
Yeah, I see Casey's still on. That was a curiosity. We were thinking about Hood Canal.
Scott?
Yeah.
Just as I tried to come alive, you changed the topic. But if you could go back to Southern residents,
no problem.
I would just like to note that, and we can see if this confirms or is agreed to by anybody else, but that this year and in recent years, they've been all together. Currently 24 JPOD members. And yes, they're spread way out, but they're basically all there somewhere in that moving mass of orcas. And if you go back, 6 or 7, 8 years, 2013, 2014, 2015, they often were so fragmented. The matrolines were coming in without any sign of any other JPOD members. J16s came in sometimes, J19s. And I wondered if they were going to ever regroup and regain that cohesion that they've had before. So it's good to see that at least they are traveling as a JPOD unit, all of them present recently. So I just wanted to note that that's a trend that I'm seeing.
Great. Interesting, Howie. Thank you. So I wanted Dave Bain's not on the call, but he has been moderating lots of Biggs candidates that where the Biggs fool the artificial intelligence into thinking that Southern residents are present. So I just wanted to flag that on August, Dave, who's got 40 years of listening experience in the area, flagged one of the candidates or one of the Biggs events as the longest series of transient calls he'd ever heard. So you can explore these annotations and the recordings, but I just thought it'd be fun to play an example of what that sounded like. So again, that's off of the Bush Point hydrophone. So that's leading to enough events, acoustic events, that we're becoming a source of Biggs killer whales annotated data that is helping some of the Canadians who want to tease apart and teach machines to separate between northern residents, southern residents, Biggs, and humpbacks. So it's been valuable to have them around more often, because we're hearing them more often than we used to, for sure. So I think you already touched on this. You already totally touched on that. I'll just erase that, Elisa. That's great. And the last thing I wanted to flag on Biggs is, since Monica's not here, she's been posting updates to Facebook, which I think are noteworthy because it's not a peer-reviewed publication, but it's a near real-time view of the occurrence data without effort correction. And I just think it's interesting that the Facebook groups are not only providing a denser stream of data, but they're visualizing it, and it's enabling a much broader community to think about why animals like the bigs or the southern residents are moving in particular places. And it happens in near real time. But I hope we can get Monica to talk a bit more about how she's been pulling data in from those sources, as well as the Pacific Whale Watch Association over on our winter call. We kind of touched on harbor seals a bunch this meeting, but I asked just briefly if anybody has additional observations about harbor seals in 2021. All right, and how about California sea lions or stellars, beyond what John already spoke about? Wow, I'm in trouble there. I'm just going to back that out. Harbor porpoises, any new thoughts there? All right, we're coming to you, John. New thoughts about humpbacks in the last year or so?
Yeah, I just added a few notes there in the agenda there. If anyone had any questions about those, just some of the ongoing research activities we've had going on this year related to humpbacks. And there's probably more I could add there, but if anyone has questions about those three points, the new abundance estimates, the Splash 2 study, or some of the tag deployments we were doing.
I guess I'll jump in a little bit here too. I'm Emily, and I was at Orca Lab for September and October, and we started hearing a lot of humpback vocalizations. And Helena asked me to start putting together a Nong-Song humpback vocalization dictionary that they can start using. And I brought this project to Scott and Val as well. I guess one thing that I have observed, especially at Orca Lab, was there are lots of bubbles in the recordings made with these Nong-Song vocalizations. So we don't know whether, and it could never be confirmed visually because most of these recordings were made during the night hours. But that would be something interesting to look at, is the type of feeding activities that are happening in both Salish Sea waters and Johnson Strait waters. I think that's also a project that could happen in the future, is maybe collaboration to put together a catalog of the whales that are most common in the area, start grouping them by gender if we have it, just to kind of get a better array of singers that we may have in the area and these behaviors associated with their Nong-Song vocalizations.
Yeah, somewhat related to that, I wondered, John, if you had any new insights from the tag data about what the humpbacks in Strait of Juan de Fuca or other inland waters might be consuming.
Yes, we were getting pretty good data on feeding behavior on the animals, and also doing some hydroacoustics at the same time. So in a bunch of the areas we were studying, there was a deep krill layer that it seemed like they were targeting inside at the Western Straits entrance. And we saw quite a bit of movement between animals concentrated in the Western Straits and then out to Swiftshore Bank and areas just offshore in that area. We haven't fully analyzed that. We did deployments in both June and early September. of these tags that gave us typically like 2 to 7 days of data of high resolution movement data and dive behavior of animals. And we were seeing some pretty dramatic differences in daytime versus nighttime diving behavior, which is something we were after, and also very kind of very heavy use of just shipping lane areas, which was one of the goals of the study with DFO. DFO purchased the tags that we deployed, and we'll be analyzing the data. So anyway, those are a few tidbits from that work. We did deployments last year as well, so I think now we have something close to 15 to 20 deployments on animals. So humpback whales.
So as Emily and I go through some of these recordings, and I don't think we've heard any bubble sounds, but we haven't been listening for those. But you have any initial thoughts on how they're foraging for those krill?
Yeah, most of the foraging we saw was deep and not in surface waters. But again, that was the Western Straits. Where are you guys picking up these? What's the location again that you're getting those bubbles down?
Yeah,
so we were seeing these in the Johnson Strait.
Johnson.
So I was just curious about feeding behaviors or foraging behaviors in the Salish Sea and seeing if there was any bubble net feeding.
Right. I do think bubble net feeding is something that there's been I think a couple of discussions going back a few decades where bubble net feeding was of course heavily seen in Southeast Alaska and northern BC, and we kind of speculated on the degree to which it might spread as a behavior southbound, because we hardly ever see bubble net feeding on the US West Coast. Anyway, how that might spread. It does seem like that's usually more targeting at least the group bubble net feeding fish aggregations, and I think that might be more of a prey in the deeper inside waters than what we were seeing closer to the entrance of Strait of Juan de Fuca. So that could be a difference between those animals further inside and those outside.
Excellent.
And I can add as well. This is Holly, with the work that we did in collaboration with Lance Barrett Leonard up in Telegraph Cove and Johnson Strait. We did see bubble netting with some of the humpbacks, but it wasn't the pattern that we've seen in other study areas. It was bubble netting along the shore, and they appeared to be feeding on, we thought maybe capelin, but it's almost like they were trying it and not quite getting it right, but it was different. It wasn't that circular pattern that we've seen down in Antarctica or Cape Cod or like they see up in Alaska, but it was more of a linear bubbling. But we did see it on multiple occasions. I don't know. I mean, we have a record of which individuals, if that might be helpful, but.
Yeah, that's interesting because on the recordings too, it does sound like they are very short bursts of bubbles. It doesn't sound like the full feeding cycle. So yeah, actually I'd be really interested in the individuals that you saw doing that. That would be really helpful. Also to help identify the vocalizations and the vocalizers. So here, I'll just put my email in the chat. Thank you so much.
Okay, sure.
And I'll just flag that I'm excited to work with Emily, who just finished at Colorado College last spring. The Happy Whale and the Fluke Book projects have really been fantastic for contextualizing what we've been hearing, because often, especially in the fall, as more and more individuals leave the area, you may get an ID at the right time that indicates which individual was making the call. So I think it's exciting. And for those of you who would like to explore what the AI has been hearing, I'll just flag that there's a dashboard view of all of the detections from the last year of testing this system. And you can search the tags for different types of things that we've heard. So those are all the bigs. Each of these is a 60-second recording. So if you want to listen to the humpbacks, you can search for humpbacks at that dashboard, which I've linked in here. So we'll report back hopefully in the winter and spring on the integration of those data sources with Emily's help. Let's see. Do you want to take a few minutes and update us on gray whales, John? Or anybody else?
I can just make a couple of comments there. I did add a couple of notes on the UME since you had that question about a UME update. So Jesse Huggins, our stranding coordinator, has been active with that, working with many of the other stranding network participants in Washington State. And I just put in, those are Washington strandings, from the big UME year of 2019 of 34. It has dropped quite a bit, 13 in 2020 and 9 so far in 2021. So those are still above normal. They're above the average, but they're well below the 2019 level. So there does seem to be a recovery from the UME. The numbers in other regions along the migration route of gray whales have also shown declines, though not seemingly quite as dramatic as what we've seen in Washington with that level of decrease. And that's still being investigated by the UME working group. And there are 3 different subcommittees of that working group kind of actively exploring, connections to ecosystem, population dynamics, or insights into kind of causes of death from the examinations that were conducted. And I think there's some good information on the context for that unusual mortality event, but maybe still short of a complete understanding or explanation for it. And it seems like the concerns are that on the one hand, the gray whale population has done really well and had reached very high levels prior to this UME, like it had prior to the 1999-2000 UME that it did bounce back from. So that plays a role, but there have been dramatic changes in the Arctic, both in terms of ice cover and some of the prey abundances seen in some of the areas gray whales feed. Many of the animals that strand are emaciated, but it's been hard to rule out all other causes, that might be contributing to that as well. So I'll just say that much on the UME.
One quick question on this. Are you hearing vocalizations in the inland waters of Washington?
Yeah, so we had deployed in the past these suction cup attached multi-sensor tags, but only this year for the first time were we using ones with good acoustic sampling. And so there's a woman starting graduate school who's been working with us, Hannah Clayton, who's been both going through the current tags we deployed and some of the past tags using the accelerometry signal to look at gray whale calls. And she has documented hundreds of calls detected on these tags of the sounders gray whales around Whidbey Island. They seem to mostly occur when they're socializing, not during the feeding periods. She's working also with Rihanna Burnham in BC, and I think that'll continue to be an emphasis we have. And I think it would be a great one to talk to you more, Scott, and I think should be interested in talking to you more about, now that she's developed a little better understanding of, gray whale. It seems like gray whale vocalizations got attention early on, many decades ago, and then in the kind of explosion of interest in acoustic detections, they seem like they were sort of ignored for a while, and now they're being caught up with. Again, is sort of my perception, but how much is that something you guys have detected or monitored?
Well, I think you've given Susan a good motivation for a Langley hydrophone, but we have not heard them on any of our efforts. But the ONC folks on the outer side of Vancouver Island and Barkley Canyon have recorded them.
Right.
So I've been mystified as to why we're not hearing them. So it's very interesting.
Yeah, no, Susan, I would definitely put a plug in, a bunch, that the one tag that I think she got something like 100 vocalization detections was just over a 1-day period. And that was an animal that was in Port Susan around Hat Island. We tagged it just off the Snohomish River Delta. And I think it was in Saratoga Passage as well. So any of those locations, maybe even something, I don't know, that could both have a good unobstructed listening path, if you will. I don't know how to describe that, but they could hear that south end and north end of Hat Island, as well as up into Saratoga Passage would be a great location.
Great. Yeah, we'll check in with you and Hannah to see where the best location is, or if we need one on Whidbey and maybe one up on Camino or off Everett or something.
Right.
Yeah, awesome.
I know we're running out of time, but I did get a response just today from Kristen about the strike that Howie, you told me about, of a gray whale, probable gray whale by Kitsap Ferry. So I just want to, oops, do that. I just want to flag that response and not lose it. Kristen. Anyways, I will dig that up while we continue to talk. But
I think that's it is interesting, and I think there is something that it would be very worth writing up. I know we submitted an abstract on some of those ferry strikes of humpback whales, and I think, you know, I think this more recent one that you're highlighting there, you know, is what at least our third case of a large whale documented with a, you know, details of where and when and speed, and so many of the ship strikes that occur between large whales and vessels, you know, you don't get the sort of documentation of, you know, what vessel going, what speed, at what location. You know, it's, I think because of the high activity and awareness of whales, the number of ferry passengers, the number of observers, the strikes when it involves a ferry tend to get better documented than is probably occurring with the much more numerous, you know, fast cargo ships that are going through our waters. But I still think there's something very interesting and valuable to be learned by the fact those do tend to get documented, you know, more than would with any other vessel type.
Right. Yeah, I know Elisa worked with some of the reporters on that one, and with Kristen. I'm not sure if she's still on.
Yeah, I'm here. I don't have any more information than what Scott has there from Kristen, but yes, no, because we did get the report from someone on the boat, and I am just looking. There was some gray whales in at that time. And
I mean, one other instructive thing from that one and the other two involving humpback whales in recent years has been that at least the first two with humpback whales, and like, you know, it's maybe not as sure because it wasn't as well documented with this one, the ship strikes, at least in the first two with humpback whales, were almost certainly fatal. And yet no carcass was ever recovered. And I think it just highlighted again that piece that, you know, especially animals that are struck where it pierces the body cavity, you know, have a high probability of sinking and not coming to the surface due to bloating, especially if they've been struck in a manner that breaks open the body cavity.
Yeah, and I know Elisa witnessed a near miss with another ferry that, you know, a gray whale was like right in its path as it was coming out of the dock, and she was on the phone with them warning them about it. But that one didn't happen, as far as we know.
yeah, and like John was saying, I remember trying to keep track of the gray whale reports. We didn't have any in the days after that hit, and there was a gray whale in Shilshole Bay area, right in that same general area the day before. So it seems pretty likely it was a gray whale, and the crew thought it was as well.
And a fatal, yeah.
Last thing last thing I wanted to ask you, really you, Elisa, about was whether I'm right in that there were gray whale sightings this fall in places I hadn't heard of them before, like from the Elliott Bay waterfront and down well south of the Tacoma Narrows Ridge and the Nisqually Reach. Were those anomalous, or are we looking at deeper incursions into Puget Sound by gray whales than in past years? Does anybody have a thought on that?
It's not unusual. John would have more info on that, but there was one whale that stayed in Oro Bay, which is South Anderson Island, for over a week, coming and going, but staying right in the bay a lot. Their community got pretty on board and were vigorously documenting it and getting into a lot of other mapping and things. It was pretty cool. But no, I think, John, don't we each year get at least one or two down in the South Sound, at least moving about?
Yeah, and I was going to say it is something that seems to go in, in periods across the years, and is associated often with some of the, elevated mortality years. We get more of these stragglers coming into Puget Sound, especially in spring, and then also another but smaller peak that occurs sometimes in late November and December. Both of those matching with the timing of the primary northbound and then southbound migration past Washington State, that you'll get those stragglers coming in. And yes, check out the Anderson Island group. Has a whole website with links about their gray whale. So they did really get into it.
It's great.
A Facebook group?
It's a website. I could try to come up with the link. I know they were, I think it was just a regular website, if I recall correctly. I'll look that up and maybe put it in the chat if I can find it.
Yeah, if you can. I'm working 2 computers, but Belen has been a longtime sightings volunteer, and she was spearheading the community down there, and she wrote up a really nice piece summarizing the whale stay, and included some of her maps, and posted it to, she shared it to us. I'll share it later tonight, but also Puget Sound. But I think she probably sent you a link too, John. Yeah.
Yep, I'm just I'm pasting it in the chat right now. That's it right there. And it was Belen that I had heard from, so you're right on with that.
Yeah, it was fun, but it was also an enormous amount of work during a really busy time. There was emails flying around, but it was fun. They did a good job.
Super. Well, there's a few more here. I invite folks to add comments in the Google Doc, but it is 4 o'clock, and I see some folks are needing to sign off. So I wanted to make sure everybody felt that it's okay to sign off. And this last section is work that we can do asynchronously. I'll just cue you via our email distribution list to approve in a very belated fashion what our plan was for this year, and start thinking about a plan for next year. But briefly back to the gray whales and the strikes, I think it's worth revisiting that we have sort of emergence of fast ferries in central Puget Sound as a new source of risk. And so maybe we can do a better job at keeping track of when we are likely to see those gray whale incursions, and flagging those periods for at least for that industry, as well as Washington State ferries. So I'll put that on the agenda for winter to come back to that.
Can I ask a question here for the baleen whale sightings? Are they being reported to the RAS system? Because the ferries use the RAS. So if those sightings are not getting into the RAS, they're not going to get to the ferry captains. And that's been something that Washington State ferries have raised with us.
Yes,
Well,
and I'll speak to that a little bit. So when I know of them and when I'm out there, or monitoring sightings down here, yes, I do input those. I presume other people on shore do as well. I don't know. And then we've been working with many government and other groups to secure more funding to help with that effort. Yeah, because we're maxed out, and so when I can, I do. But we're very short-staffed on the sighting network, so we're trying to get more funding to help with that.
And we also have for decades worked directly with Washington State ferries, and Elliser calls them directly when she's aware of southern residents, or like the day there was a gray whale right behind the ferry as it was backing out. So we also, yeah, are in touch with them directly.
Frank, quickly too, I know one of the humpback sightings was, the humpback that was being observed by a whale watch boat at the time it was struck. And they also made attempts to relay by radio real time, the things that are happening even faster than something like the RAS can take care of in terms of immediate danger. There's a whale right in front of your boat.
And Frankie, I could add on the acoustic side that we have definitely offered to Oceanwise to access our automated and real-time detections. Early on, like as of a couple of years ago, they weren't interested in acoustic data because it wasn't geographically specific enough and didn't have directionality. If we were properly funded, like we could probably provide them with an actual movement, like a direction of not just a location of detection distance around one of the hydrophones. So I think there's probably many opportunities to integrate better with RAS. But to some extent, the ball is in the Canadians' court, at least in terms of ingesting acoustic detections from Orcasound. A great question. I think I saw Todd's hand up. Did you have a question or a comment before we stop recording, Todd? Looks like maybe his hand's down. So yeah, so why don't we're 5 minutes over now. So if you wouldn't mind, Katrina, just stopping the recording, and I'll make sure we follow up with the rest of the agenda through some emails.